Eita,
Atención, estas son...
El sistema de alerta sísmica se procederá a evacuar el auditorio Julián E. Villarreal. El inmueble cuenta con 2 salidas de emergencia laterales ubicadas a la altura del escenario y una salida frontal en el acceso principal. Le pedimos identifique la más próxima a usted. Si se encuentra en la parte inferior del auditorio, la evacuación se realizará por las puertas laterales de emergencia. De ubicarse en la parte superior, deberá salir por la parte frontal a través de los pasillos laterales. En caso de presentarse un movimiento sísmico, replegarse a los pasillos laterales. Una vez terminado el sismo, se procederá a la evacuación general del inmueble por las salidas de emergencia hacia los puntos de reunión. Se le solicita conservar la calma y no obstruir pasillos y rutas de evacuación señaladas.
Gracias por su atención.
tá voltando?
Aqui agora. É porque o rapaz mostrou, né? Que tinha que ver se tinha ficado ok a tomografia. Aí eu fiquei para ver. Aí deixa eu ver como é que ficou o jeito.
Fechem o microfone.
Ficou?
Ficou.
Oh, que bom. Eu tô aqui no evento. Não, tô no evento aí abrindo lá do Zoom aí.
En caso de la activación del sistema de alerta sísmica, se procederá a evacuar el auditorio Julián E. Villarreal. El inmueble cuenta con 2 salidas de emergencia laterales ubicadas a la altura del escenario y una salida frontal en el acceso principal. Le pedimos identifique la más próxima a usted. Si se encuentra en la parte inferior del auditorio, la evacuación se realizará por las puertas laterales de emergencia. De ubicarse en la parte superior, deberá salir por la parte frontal a través de los pasillos laterales. En caso de presentarse un movimiento sísmico, replegarse a los pasillos laterales. Una vez terminado el sismo, se procederá a la evacuación general del inmueble por las salidas de emergencia hacia los puntos de reunión. Se le solicita conservar la calma y no obstruir pasillos y rutas de evacuación señaladas.
Gracias por su atención. Atención.
Las neurociencias han cobrado una relevancia especial en el mundo científico en los últimos 10 años. Los países desarrollados han realizado una inversión histórica para tratar de entender los misterios del cerebro y volcar esos nuevos conocimientos a la prevención, los tratamientos y la cura de las enfermedades cerebrales.
Al mismo tiempo, se han desarrollado nuevas tecnologías y se han promovido cambios en la educación, la cultura, la economía y otros ámbitos de la vida de sociedad.
Latinoamérica está preparada para enfrentar este gran desafío. Estamos dando los primeros pasos para sumarnos a este emprendimiento global y ser también protagonista de los cambios históricos que se están gestando.
Uno de los retos más importantes de este tiempo es desentrañar los códigos secretos que hacen funcionar al cerebro generando nuestros pensamientos, deseos y acciones. Múltiples alteraciones y enfermedades relacionadas con el cerebro, como por ejemplo, adicciones, violencia, trastornos neuropsiquiátricos, del desarrollo y de la cognición social, afectan negativamente la calidad de vida de los individuos. Además, provocan grandes pérdidas en la productividad y en la economía de nuestros países. Debemos incurrir en gastos enormes para sufragar los tratamientos y tratar de dar respuesta a los problemas que estas dolencias implican.
Al mismo tiempo, los nuevos conocimientos y desarrollos tecnológicos, los beneficios y transformaciones que podemos imaginar a partir de un entendimiento más profundo e integral sobre el funcionamiento de nuestro cerebro son enormes.
Sin duda, en Latinoamérica estamos en condiciones de establecer centros de referencia en ciencias del cerebro. Podemos establecer plataformas de cooperación que permitan aumentar y optimizar el acceso al conocimiento y la tecnología. Asimismo, debemos aumentar el flujo de investigadores y estudiantes entre los países que conforman las diferentes iniciativas cerebro, agregar aspectos culturales y soluciones particulares para los países de nuestra región. Hay que mejorar aspectos éticos, bioéticos y de igualdad de género en ciencia. Debemos establecer un plan estratégico de desarrollo de ciencias del cerebro que incluya la complementariedad de capacidades y necesidades de nuestros países. La educación es esencial en todo este proceso.
La misión de la iniciativa Cerebro Latinoamericana es potenciar y avanzar en las investigaciones del cerebro
a través de la colaboración y el intercambio de conocimientos, uniendo las diferentes aproximaciones y enfoques a nivel latinoamericano para expandir el alcance de nuestras investigaciones y así producir y difundir nuevos conocimientos y sus aplicaciones científicas para el beneficio de nuestros países y del mundo. Nuestra visión es la de alcanzar un conocimiento profundo e integral del cerebro que nos conduzca a mejorar la calidad de vida de las personas en los diferentes niveles, tanto individual como social, conociendo nuestras peculiaridades a nivel de Latinoamérica, pero al mismo tiempo reconociendo los puntos que tenemos en común a nivel regional y mundial.
La iniciativa LATBrain tiene como principal propósito el identificar prioridades comunes relacionadas con el estudio del cerebro en la región. Sin duda alguna, se requiere el intercambio de ideas con el propósito de oportunidades y retos que puedan ser alcanzables en corto y a largo plazo en nuestra región de Latinoamérica. Es necesario el establecimiento de redes y colaboraciones estrechas entre académicos y académicas, científicos, científicas, industria privada y todos aquellos que estén relacionados con el estudio del cerebro, con su tratamiento y todos los profesionales que estén interesados al respecto. Asimismo, es necesario estimular la colaboración en nuestra región y el establecimiento de nuevos programas que nos permitan alcanzar los objetivos de esta iniciativa.
El plan estratégico de la iniciativa Cerebro apuntará a entender cómo se desarrolla y funciona el cerebro normal.
Pretende comprender el origen de las diferentes patologías sobre el cerebro y utilizar la información que tenemos del cerebro para mejorar la salud mental en la región.
De esta manera, se aportarán insumos que nos permitan mejorar la educación, el bienestar de la población y el desarrollo económico y social de la región.
Los participantes de la iniciativa Cerebro Latinoamericana reconocemos que entender el cerebro es un esfuerzo tan grande y complejo que ninguna sociedad o país trabajando solos será capaz de enfrentar este desafío que implica medir, mapear, modelar, visualizar, simular, entender, imitar, diagnosticar y sanar el cerebro. Nos comprometemos a organizar a nuestros países en esa dirección para de manera conjunta alcanzar nuestros objetivos que representan necesidades comunes de todos nuestros países.
Invitamos a todos los actores de la sociedad interesados en participar en la iniciativa Cerebro Latinoamericana. Invitamos a apoyarlos, adhiriéndose a ella y también participando en las diferentes instancias de trabajo. Para el desarrollo y fortalecimiento de esta iniciativa, no solamente están invitados a participar las organizaciones científicas y académicas, sino también empresas e instituciones civiles que estén relacionadas a los temas de investigación y, ¿por qué no?, entidades financiadoras que finalmente tomen decisiones políticas a este nivel en cada uno de los países. De esta manera, el impacto de LATBrain va a ser mucho mayor y esta iniciativa llegará a las personas y a la sociedad en general.
El trabajo conjunto de nuestros países abre una nueva oportunidad de desarrollo científico en la región y la posibilidad de avanzar para mejorar la salud y el desarrollo socioeconómico. Únanse a nosotros en este gran reto. El éxito de la iniciativa depende de la participación de todos.
pasamos a la presentación, por favor. Hello, good morning, everyone. I'm very happy to start this summit, the first one of the Latin American initiative, BRAIN initiative. I'm going to talk something about this initiative. It is well known that the brain, it's a very attractive organ since the ancient times, and Latin American is not the exception. I'm showing here a school of a person who had a brain surgery, but in ancient times, and this person survived. Therefore, in Latin America, we have a very good experience in neurosurgery of the brain. In 2019, a group of scientists were together in Uruguay in order to start and discuss how to start the Latin American Brain Initiative. Since then, we have had a lot of activities that I'm going to talk something. The main vision and vision of this LATBrain is to make strong collaborations through the region in order to improve the health brain and also to find new therapeutic strategies to treat brain disorders. It is obvious that at present time there are important alterations that affect the worldwide. More of them are focused to neurodegenerative disorders, and we have to be aware about this situation, especially after COVID-19 pandemic. We were concerned about the necessity to establish good collaborations through the region. We started looking for identity, and first of all, we want to get a good logo and good slogan. We organize a competition in order to get the best logo and slogan that could represent LATBrain. And these are some examples of the drawings we received. And the winner was this, in which Latin America and Caribe areas are connected by different types of neurons. And also the slogan was Un cerebro conciencia y conciencia latinoamericana. In other words, a brain with science and Latin American conscious. This is our logo and slogan. Since then, we have been organizing the different symposiums.
Para los que estamos en línea no se escucha.
Excuse me? Yes, problems? The first symposium was focused to present the different efforts and consortiums through the region, focused to evaluate neurodegenerative disorders in the region. In 2023, we organized another symposium focused to evaluate different problems in the brain of immature patients. And now we are celebrating the first summit. And this is a very important, strong effort supported by different associations. First of all, I want to acknowledge the staff of the Center for Research and Advanced Studies that during this year we are celebrating the 25 years of establishment of this unit. Also the Mexican Society of Physiological Science, the International Brain Research Organizations, and also Psychopharma. It is important to let you know that at present time, LATBrain has been considered as a member of the International Brain the International Brain Research Organization. And also, we are members
member of the International Brain Initiative. This international initiative congregates the most important brain initiatives worldwide. Here it's the
recording in progress. Recording in progress. Recording in progress. Recording in progress.
Here is the logo of LATBrain, and you can see that the different initiatives are in this international initiative, but what we can give to these international initiatives as region? Well, we can give different resources. No está avanzando. For example, in the region, we have natural models like alpaca. It's a natural model of an animal that has homeostatic mechanisms to avoid the hypoxia. We have Proechimus guyanensis, a rodent that is found in the Amazonia. This rodent is high resistant to epilepsy. Also, it is well known that in Latin America we have traditional medicines, and in addition, all the population of Latin America has a specific gene composition. These are some of the resources that we can share to the world. At present time, we are very active showing all the efforts that we are carried out in the region. For example, in our website, you can find the most recent publications published in international journals. Also, we are doing education and audits of the respondent persons that participate in the region. The next one. Most of the people were between 25 to 65 years old, were very active, and most of them were focused on PhD degree, but also there were undergraduate, graduate, medical professors. Most of the persons that responded to the survey were basic scientists, but we are very happy because medical doctors also were interested in this kind of activity. Next one. The areas that the responders were working on were mainly in central nervous system disorders and also cognition and behavior. And this figure shows all the techniques that are used in the region, going from the behavioral test to artificial intelligence. Obviously, we ask what are the most important needs of the regions or the country, and the most important answer was funding, obviously. The priority areas of the country were focused on neurodegenerative disorders, psychiatry disorders. Therefore, we have professionals that are aware about the most important problems in this region. The training of human resources of the country were identified like good, excellent, and good. And now we have different activities focused to disseminate the education tutorials that are present in the region in order to make strong this region, in order to work together and get new treatments. And it's very nice to show you that at present there are different participant agents through all the region from different countries, and we are waiting for you, for joining you. Finally, we are very happy to stay here it is very, we know that we are living in a very hard time. However, we have to work together. And I want to invite Dr. Monica Lamas in order to say some words about these aspects. Dr. Monica Lamas is the director of the Center of Aging Research. Thank you.
Thank you very much. On behalf of Dra. Claudia González Espinosa, the director of this campus, I welcome you to our grounds. I am the director of the Aging Research Center that you will visit later. And we are very happy to have you here. We will host you for your lunch probably. And we are a recently created group with researchers from different disciplines, from biology to chemistry, from medicine to genomics. And we use different experimental models, like in the Department of Biology, stem cells, yeast, hydra, rodents, humans. And we evaluate different systems, like the muscle or the immune system, or of course the brain. So that's why we are also very interested here. And we believe we are a recently formed group, and we believe that our strength comes just from that, from the collaboration, from working with one another, building bridges, talking about different things, in our case, about the same subject, the complex process of aging, but from different perspectives. And we believe this is our strength, and we believe also this is the most important thing that we can give to our students, this rich environment and this collaboration, this sense of community. And I believe the aim of this meeting is exactly that, to form a community, to build bridges, to facilitate collaboration. So for that, we are very happy to have you here, to host you here, and we wish you the greatest success. Thank you very much for coming. Thank you very much for joining, everybody online. And welcome, and if anything, you need anything, please ask our staff. Thank you very much.
Yeah, Good morning, everyone, and welcome to the first symposium of this important activity. Today, we gather to reflect on one of the most vital aspects of our future, brain health across the life course, and particularly within the Latin American context. In this symposium, Making Brain Health a Priority in Latin America, strategies to assess early child development in ways that truly reflect our local realities will be explored. Next, we will turn our attention to aging brain, examining healthy and diseases aspects. Finally, it will be addressed one of the greatest challenges facing Latin America, the growing impact of neurodegenerative disorders. Our hope is that this meeting is not only sparks the new knowledge and discussion, but also inspires action so that together we can strengthen brain health across all generations in our region. It's my great pleasure to introduce our first speaker. Dr. Laura Sanchez Vincitori is a professor at the Universidad Iberoamericana in Santo Domingo, República Dominicana. She leads the Institute of Applied Neuroscience, and she is the country's representative in this LATBrain initiative. Dr. Sanchez will present our strategies to evaluate early child development in the Latin American context. Please join me in warmly welcome Dr. Sanchez.
Good morning. Thank you so much for being here. So while we wait for the presentation to show, I just want to start by saying that we don't change what we don't measure, and that's why it is so important to measure childhood development. I'm going to check if this works. If it doesn't, I'm going to tell you when to move the screen. It's working. So we have neuroscience and childhood development. I mean, it's kind of obvious right now, like most of brain changes and growth occurs during the early stages. The prenatal stages, for example, start 2 to 3 weeks after conception, and we have neurogenesis and neuromigration and synaptogenesis. And there is a huge influence of environment in general development, especially stress, nutrition, and alcohol. When mothers go through difficult times, that has an impact on childhood development. So we have the developmental lifelong health and disease origins, and sometimes it has to do with early experiences and early environment. So the same thing happens in early childhood. Brain triples in size during the first 2 weeks. We have sensitive windows, which are moments in development where some abilities should be acquired, otherwise it will be very difficult to acquire them later on. Especially with language, executive and social and emotional development. And what research has shown is that children who are well taken care of usually do better than children who live adversity. So positive care equals growth. And also the early stress has a big influence in development in terms of the HPA axis dysregulation, the stress regulation, hypothalamus, pituitary, adrenal axis. And of course, it increases the risk of anxiety and depression, addiction, and so on and so forth, including disabilities and difficulties in learning. And now it's not working. Can you go to the next one, please? Thank you. So then, the previous one. So what you see here is part of a study that we conducted a couple of years ago with the MIX survey, which is the multiple indicator cluster survey that countries conduct every couple of years. I can't recall. So what we did is that we created 2 different models that predict childhood development. A model that had to do with only socio-demographic variables such as sex, age, maternal education, and socioeconomic position. But we also contrast this with another model that included other psychosocial factors. And psychosocial factors are those that you can actually modify, like discipline, like stimulation at home. So what we found was, of course, that socio-demographic variables had a big impact in childhood development, especially of course, age and sex and family socioeconomic position. The one that had more weight was family socioeconomic position. But when we put the psychosocial models into the model, those variables that had to do with socio-demographic aspects would just reduce the effect just a little bit. It's a little bit of a buffer that these psychosocial variables have. Those include good stimulation, the child goes to a child care, and the child does not receive negative or violent discipline. So we have a link of the things that we do with our children and the way they develop later on or while they're developing. Let's go to the next one, please. So again, what we don't measure, we can't change. We have a lot of challenges in Latin America in order to change this measurement thing. It's not easy to have robust and large measurements. The first thing is some instruments might not be applicable to the context. I mean, it is true that some part of development is biological, it's biologically determined by genes and codes, etc. But there are cultural factors that kind of shape the way this development is going to go through, and it is really difficult to measure, to use measurement from a different context into the Latin American context. So we need to adapt those measurements, and we need to make sure we're measuring what we're supposed to be measuring and not some other cultural manifestation of a different context. We have lack of funding and trained personnel. Childhood development is not easy. It requires very highly qualified personnel. And again, need for large and representative data. It's difficult to get the logistics to create an evaluation for representative samples in any context. Imagine our own Latin American context. And again, household surveys, which are the ones that are usually conducted in our countries with really robust methodologies and with representative samples and probabilistic samples and power analysis, etc. Sometimes those are not enough in terms of measurement. The childhood development measurement has 10 items. It's like a picture that you took with a, I don't remember, like 1998 cell phone that never took pictures. It's blurry. So you can, you know some general aspects of development, but we can't see the details. And that's, you know, it's very good for decision making or for knowing what's going on in different contexts, but it's not really good at telling us what's going on with actual childhood development. So what we've done, this is work from the Dominican Republic. We created the Dominican System for Measuring Early Childhood Development. It's CIMEDIT for its acronym in Spanish. And it's a memorandum of understanding with 3 institutions. One is UNICEF, representing the international cooperation. The other one is the Universidad Iberoamericana, it's the university I represent right now. And INAIPI is the Instituto de Atención Integral a la Primera Infancia, which is the government agency that is in charge of providing and coordinating services for early childhood. Like the success of this program is due to the fact that it was not a single institution working on this. It was not just academia, not just international cooperation, their funding, and not just the government. It was the orchestrated work of each of them. So what does it do? Can you go to the next one, please? This is a little bit unreliable. Can you go to the next slide, please? There you go. So CIMEDIT is an electronic platform for screening for childhood development. We chose to do an electronic platform because they serve 200,000 children. Having paper evaluation would have been difficult. And also to compensate for the challenges of needing specialized professionals. This electronic platform actually tells the evaluator what to do and when to do it. So if you want to know the development of a person, that person is subscribed to the database, and the database connects to the electronic app and decides where the child will start the evaluation, and the evaluator just needs to follow instructions. So the child can hold a ball, and you give the ball to the child, and they say yes or no, whether or not the child passes or fails that specific item. It operates via a mobile application that can function offline and synchronizes when there's internet available. So it depends on technology, but it does not depend on connectivity. And it is designed to generate high precision data for a lot of things, like evidence-based practice, but practice with the child in the classroom, but also at the center level and at the country level. Part of the international requirements is to report every year the percentage of children that are on track with development is de los ODS. And since before we didn't have a measurement to tell you this data, but now we do. So we generate high precision data for everyday life and also for international and national reporting of data. And the most important thing is we're able to identify children who are at risk for developmental delays and facilitate a multidisciplinary care. It's not that we do not have the multidisciplinary care as of right now, but it is in place, and we can identify these children, and we have a referral network to send children who really need support. And one of the most interesting things is this is administered by INAIPI personnel, which means that the tool was born at INAIPI for INAIPI, and they are using it. We did not have to go through a process of convincing anybody to use these things because we created them as a group, and it started where it was the end user was the institution that created this. Everything is in IP servers. We just have access to the data, and as researchers, we just evaluate the data, the quality of the data. We publish papers, but they are actually using it from the day one, and we did not have to wait a single moment because they started it. Let's go to the next slide, please. There we go. So we measure childhood development in 4 different domains. It's based on other scientific work. We uses a model that the Malawi Developmental Assessment Tool, which is based on international standards for measuring childhood development. We measured gross motor, fine motor, language, communication, and socio-emotional development. Each subtest has 33 items, or 34 in the case of language, and the child does not have to complete the whole evaluation. So one of the challenges of evaluating a child is that they grow. So you can't ask a child that is 2 months the same that you're going to ask a child that is 4 years. So this dynamic nature of evaluating childhood development has to be taken into consideration when designing this test. What it's usually done is that you calculate the age of the child and you start by the item in which there's a 90% chance that the child will pass that item, and then you go back 3 just to see if there is a developmental delay there, and then you move forward with the test until the child does not pass 3 consecutive items. So that's why we have a lot of items, but it's because we're evaluating children from 45 days to 5 years, and at the end we end up for each subtest evaluating around 8 items. Let's go to the next slide, please. Next slide. Thank you. So this is a study we conducted in order to make sure that we had it very fine-tuned. As I said, we have to start by the item in which there is a high probability of pass. So in order to do this, we did a pilot with all the items on 948 participants just to detect the probability of the child passing in a time range of age. So we did a logistic regression that predicted whether or not the child would pass, and to see if age predicts this, but what age range would predict that item. So we have from 25 to 50% of children do this item at this age, 50 to 75, and 75 to 95, and that way we organize our items. We originally took the Malawi Developmental Assessment Tool, but right now we have the Dominican version of this, so we have the items organized by difficulty, which is very important when you have this type of evaluation. Next slide, please. The next slide. Thank you. So what's next? So what are we doing with this data? So we already saw in our data that's about to be published the relationship between practices, parenting practices, and development. We already have the developmental variable already tested, so we can go back to their centers and talk to their parents to see what they do at home. So we have that, parenting practices and development, violent discipline and its negative relationship with development. We have that. We can also go to their centers to evaluate parents. Another thing that the INAIP does is home visits, and they have an electronic platform to do these interviews. Any interviews we want to change, we can add a couple of questions, and since it is synchronized to the same database, we can very easily get data from what happens at homes and the developmental outcomes. We already published a study, and just because we had the database. So we combined that developmental database from one of the pilots with socio-demographic information or any questionnaire that they used when they entered the service, and we were able to determine the relationship between a child who was breastfed and their developmental outcomes. And we published it in a high-ranked journal. Right now we're working with cultural consonance, which is a very interesting concept in which cultural consonance is how much do you approach whatever your culture believes is a good life. A good life in the Dominican Republic is not the same as a good life in Mexico and a good life in Switzerland because they're different contexts. So there's a way to operationalize this and quantify this, and we're establishing the correlation between that and childhood development. Just because we already have the childhood development variable being measured for important things, we have the chance to study what happens with the culture, what happens within parenting behavior, etc. So this work has been financed by the National Science Foundation in the United States, and we're working with Tulane University to advance not only into the developmental sciences, but also anthropology. And right now we're trying to go into the underpinnings, neural underpinnings of development, and we have a proposal under preparation to study self-regulation and cultural consonance, since there's a lot of work saying that self-regulation is related with vulnerability, and these people come from very vulnerable situations, which is one of the main requirements to get into these programs. And again, we can do secondary data analysis. This is the first time we're working with a huge data set of 4 data points and at a longitudinal study with 26,000 children. That's a lot. We have 2-year longitudinal data of development, and we're just exploring to see what we can find. So what we're doing right now is just to see if there is this thing called the summer slump, which is when children go on vacation, they don't grow as much as when they are in school. It is an obvious phenomenon, but it is the same. This is the first time we've been able to show this with data from 26,000 participants, and it's a longitudinal data, so it's the same child. So when you see that from November to May 2024, the growth was big, but when they went to summer break, the growth is not that big. They always grow because it's development. Children do grow, but then when you compare it to the end of the next year, the growth is larger. So we're publishing this probably in next couple of months. The other thing that we can do now that we have this platform is that we can test new technologies. Some parents that come, like most of the parents that come to these services, they do not have good reading comprehension skills. So if you want to have large data collections just to get statistical power and to be able to explain a couple of phenomena, you need a lot of data. And it is not logistically possible to interview 1,000 mothers because you need a Likert scale. So instead we're using auto-administered Likert scale questionnaires using technology. It's a technology that it's usually used for reading evaluations, but we found that there is a little feature that allows you to record the question and add buttons that are very intuitive for people, and people can just press their buttons as they go, so we can get large data for parents as well, not only for children. So let's go to the next slide, please. There you go. So we have an integrative evaluation framework that not only combines psychometric rigor, multi-dimensional measurement of development, but we have multiple institutions working on this, and each of them working on what we know best. Academia knows data, international collaboration, cooperation, they're like managers and people who actually have to serve children. So let's go to the next one. Okay, so how does this advance neuroscience? My advisor used to say that the first study first. So we need to describe, we have to have clear definitions of what we are measuring, to have good research questions to see what's beneath the surface. And therefore, without behavioral data, the one that we have here, it is incomplete, the incomplete picture to go down into the brain, down into what's going on. But that is our goal, and like we just set the first steps nationally to make sure we know what to do with these children. So we are working in parallel. The children are taken care of. There is a pathway of care for the child that has been determined that has a developmental delay. That person has a way to go. We are contacting, we have a repository of activities for each developmental stage, for teachers, for parents, and we're using technology to make sure to create interventions that are based in evidence, and also a local evidence that can tell us what to do with our own population, and this has the possibility of transferability. Thank you so much.
Please stay here because maybe we have a question for Dr. Sanchez. Do you have any questions? No commenters? No? If not, thank you very much, Dr. Sanchez. Now we have our second talk. It is a true honor to introduce Dr. Rainer Gutierrez Mendoza, a distinguished researcher from the Aging Research Center in Mexico. Dr. Gutierrez will share his insights on the aging brain in health and disease. Please, Dr. Gutierrez. Thank you.
Thank you for the invitation. Thank you, Dr. Luisa Rocha, for the invitation. I'm going to change the title of the talk because I study actually food intake and neural control of appetite, not aging by itself. but I do neuroscience, okay? So, so I'm going to talk about the logic of hunger and the neural control of appetite. Let me see if this works. Can you help me with the next one, please? Yeah, okay. Well, hunger is not a stomach growling, it's a prediction. Let me explain what I mean with this. So probably what we know about hunger is not really accurate. But in 2010, Dr. Jeka Ponte at Janelia discovered these hunger neurons that are AGRP expressing cells in the arcuate nucleus of the hypothalamus. What they found was that activating these cells with optogenetics produce rapid and innate food intake in sated animals. So this is very interesting because these animals, here we go. So these animals are not hungry, but when you activate these cells, the AGRP neurons, the animals start eating for as long as these neurons has been activated. Once they stop, then the animal stop eating. So that means that we can be hungry and sated simultaneously. Otherwise, this experiment will not work. That means that the satiety system is composed or is embedded in a different neural circuit, and these neurons are the hungry neurons. Okay, can you help me to put the movie, please? This. So now we have techniques to see these neurons, these hungry neurons. And what they found is that these neurons respond more to hunger and less to satiety state. Let me see if the movie plays. Can you help me to play? Thank you. Okay, so you can see that these neurons are very active when the animal is hungry and not active when it's sated. So this animal is hungry, and when they put food, just by watching the food, look, it's just watching the food, and the neural activity of these neurons is shut down. It's inhibited just before actually the animal is eating this food, okay? So how is it possible that these neurons are the hungry neurons, and when they see food, familiar food, they inhibit their activity, right? So what exactly these neurons are encoding? How is the hunger neuron signal encoded in the brain? So they later discover that if the food is novel, like for example this gelatin has no calories in it, these neurons show an inhibition, but this inhibition is very slow inhibition, because it's in the first contact, okay? It has calories, so this inhibition is because the neurons also respond to nutrients in the gut. When the nutrients are metabolized, these neurons maintain an inhibition, but this inhibition is very slow. So let's see what happened when in the second time that they see the same gel with calories. So you can now see a rapid inhibition, just as you saw in the movie, okay? That means that these cells learn that this food has calories, and they predict that it has calories in it. So just by watching it, it will show a rapid inhibition and maintain this inhibition constantly. But what happens if we remove the calories from the food? What we see is that it shows the prediction, it must have calories, but in reality it doesn't have, so there's no nutrients on the gut, so the inhibition disappears. And the next time they learn that this food has no calories on it. So that means that these neurons are learning in each trial if the food has calories or it has not calories in every single trial, right? So that's why in the Costco, they give you a sample, right? If it has calories, then these neurons will learn that the next time that you see it, you will show inhibition of these hunger neurons. The opposite happens if we start without calories and then with calories. Okay, so in summary, these hunger neurons, what they do is when they encounter a new food, the first time, if it has calories, they will show a slow inhibition of its activity, and in one single trial, that's why the samples of the Costco, they will learn that this food has calories. The next time that they eat this known food, it will show a rapid inhibition that will maintain. Also, this neuron responds to nutrients directly. If we infuse nutrients to the gut, like glucose or protein or fat, doesn't matter, they show a slow inhibition, and they respond also to sensory satiety signals like CCK, GLP-1, and other signals, including Ozempic or semaglutide. Okay. All right. So that's for the hunger neurons, and that's the logic. So if that can tell you that hunger is actually a prediction. These neurons are learning which food has calories, right? And if you activate these neurons and put non-caloric and caloric food, they will only eat caloric food, all right? But there is other systems in the brain that makes you eat, not because the calories, just because it's palatable or it's because it's sweet. It doesn't matter if it has calories. That's called the hedonic feeding system. What are these neurons? I don't know if this has happened to you. I hate it, right? Ever. Like the guy that made this decision in the supermarket is a genius. Absolutely. The only thing that they have is how many candies they sell. And just with that outcome, they hack the biology of the system. Because I will try to convince you that there is some cells in the brain that actually responds more to palatable food when the food is nearby you. Okay? Let's see. So these neurons are in a different part of the hypothalamus, in particular in the lateral hypothalamus. This region has been involved in food intake classically since the 50s. Basically, if you lesion the hypothalamus, here's a representation of this brain region in the mouse brain. It can decrease the food intake and induce weight loss. But if you stimulate electrically this region, the animal will start eating whatever is nearby, and it actually promotes obesity. But we don't know exactly what cells are being activated using this electrical stimulation. Until recently, we can know the content of the cells and the genes that they express using sequencing data. So they discovered that there are at least 4 types of cell types that compose the lateral hypothalamus. In particular, these 2 cells are the more representative, the GABA and the glutamatergic cells, okay? And these 2 works in opposite directions. These are excitatory, inhibitory, but this promotes reward and induced feeding, and these neurons promote aversion and stops feeding. We have the genetic mice, transgenic mice in the lab to study these cells. Basically, I propose this system like a seesaw, where the GABA neurons are active, the animal go and eat whatever is palatable and nearby. And it's also rewarding. The animal likes to self-stimulate the activation of these cells. The opposite is for the glutamatergic cells. Okay, can you help me with this video, the left and then the right? If you can do it simultaneously, it's okay. Okay, so here we have a mouse that has sucrose on it, and we will activate the GABAergic neurons. You can see that it will take a little bit of time for these cells to respond to the light, and then approach and start drinking sucrose. Once that they approach and stay there, you can see that the head is pulled out. And when the light is active again, the head is in, right? And keeps drinking for hours and hours and hours. The opposite, if we inhibit these cells, can you help me with this video, please? The right. This is a hungry mouse. that is drinking sucrose nicely, but if you inactivate these GABAergic neurons, the animal is not able to drink one single drop of sucrose, even if it's hunger, okay? So it has a bidirectional effect. And the opposite happened with the glutamatergic neurons. Again, can you help me with the left and then the right? So the glutamatergic neurons are the opposite. So these neurons are, this mice is water deprived, is drinking or is eating actually a solid high-fat diet, but when these neurons are activated, the animal stops eating. And the opposite happens when we inhibit the glutamatergic cells. That's why we say it's a seesaw system. So this animal is sated, is not interested in food, but once that we inactivate the glutamatergic cells, they go directly and eat the high-fat diet pellet. So it's very nice, the optogenetics, right? So you can control the behavior in pleasure. Okay, so basically this is the configuration for satiety when the glutamatergic cells are overactive and the GABA inactive, and this is the configuration for hedonic feeding. These neurons are not hunger neurons because they basically, if I put any kind of food, they will eat the more palatable, the more sweet food available. It doesn't care about the calories, these cells. Okay. What else they do? So these neurons actually, Aquetzali and Jorge asked these questions, and what they do was a simple experiment where they put sucrose in a brief access test where the animal can drink for 7 seconds one of these solutions, water, 3% sucrose or 18% sucrose, and let the animal to leak. Then let the animal leak. So this is the transgenic mice. Basically what happened is that in a normal mice, what happened is they like more the high concentration of sucrose, so they leak more. The leak rate is higher for 18% sucrose, lower for 3%, and even lower for water. Okay, this is called palatability. So if we activate these GABA neurons with water, surprisingly, the leak rate increases even more than sucrose, suggesting that they like more water than this high concentration of sucrose. But now we can activate only with 3% sucrose, and you can see now that the leak rate increases selectively for that concentration, and it's even more pronounced, the effect when we activate with 18% sucrose. Okay, so now we can actually see how these neurons respond when the animal drink different things like empty zipper, water, sucrose, or 18%. We use a technique called fiber photometry to do this. And in yellow, you can see more activation, and in blue, less activation. Here is the GABA neurons, and here is the glutamatergic neurons. So what you see is that these neurons are activated even by just licking an empty zipper. Just by moving the tongue, these neurons fire more. And the same for water, for 3%, but for 18%, they fire like crazy. That means that they respond more to the most concentrated concentration of sucrose that they know. In the opposite, the glutamatergic cells are inhibited by the high concentration of sucrose, again showing this seesaw activation. Okay, let me see. Can you play the video, please? Here we go. But we also have in the lab the microendoscope, so we can introduce a green lens and see directly these GABA neurons. Each of these flashing dots is actually the neurons, the GABA neurons responding. So you can clearly see that when the animal is drinking an empty zipper or 3% of sucrose, no response of this neuron. But when it drinks 18%, boom, you can see a high activation of these cells. So we found that these GABA neurons, they have a group, a particular group. Not all the GABA responds to sucrose, but there is a particular group that likes sucrose and respond more to this high concentration of sucrose. So in a physiological way, these neurons respond more to the most palatable and sweet solutions. Now we can play with the the site where we activate these GABA neurons. So if we activate the cells in the middle of this open field, the animal go and self-stimulate because it's rewarding, and eats basically whatever it faces in front of it. But now we can move the stimulation site to the pellet, to the chocolate pellet, and see what happens. So it starts spilling all over and eating some of this. So what will happen now if we activate the GABA neurons, but the near stimuli is the chow food, which is like salads, right? Like healthy food. So if the salads will normally activate these GABA neurons, we will eat a lot of salads. Unfortunately, these neurons are activated by sucrose, right? Okay. So the conclusion for this second part is that these GABA neurons induce the consumption of the nearest and most palatable food available. Okay, so the next time that you are nearby candies, you know who to blame. This. All right. Recently, we have faced this problem, like how to study feeding behavior for solid food, because this is a really complicated problem. We have glycometers that can report liquid intake with a high precision, right? So every time that the mice move the tongue, we can record these movements and know exactly how much the animal is drinking. So we have a perfect setup for stimulating with liquid foods. And that's why most of the people actually study liquid intake. But is it the same to eat Ensure than actually eat an ice cream, right? Probably not, right? So to study that, we need to develop better techniques to study the brain and to have the precision. And the problem of these techniques is that are really, really expensive, particularly for Latin American researchers, right? And even for the US researchers, these equipments are really expensive and not precise enough to detect the feeding intake and the microstructure of feeding intake of solid food. So that's why we developed the crunchometer. Let me introduce the crunchometer. This is already a preprint. It's under review in eLife, so you can actually check the publication, and it has all the code and the software that comes from this and all the instructions to set up in your own lab. It's an open source and low-cost method to study feeding intake, and it's completely available to whoever wants it. Even for companies, it's available. Okay. And what is the idea of the crunchometer? One day I said to one of my students, what if we put a microphone to hear the bites of the mouse? And he said, oh, that's fantastic. So he ran away. We bought the microphone. He built this fear system. And then he left for a postdoc at Duke University. And the project was forgotten for 4 years. Nobody used it. And it was like abandoned completely. Later, I received a call from one of my friends, Diego Bojorquez at Duke University, and he said, oh, I have a paper that one of the reviewers want to see feeding intake in real time, okay? But it's rejecting the paper. We have 3 years trying to publish this paper, and it's not working. And I say, wait a minute, I have something that might interest you. Then I show him the crunchometer. And he said, oh, this is fantastic. He sent a student. We set it up, and I wrote the code, and it works. So the paper got accepted, actually, in Nature. So that's very nice. Okay, so then we decided to do a paper for the method, because it was so nice, this method to measure. And the idea is very simple. So we have the microphone, we have the spectrogram, we bypass the activity, so we threshold it, everything that crosses that threshold, that we'll consider a potential bite. It could be an artifact, someone clapping or someone talking, but it could be a bite, all right? So once that we have these, so we have the snippets, we build a snippet so we can see each single bite. So we have the evidence of this is really a bite. It's not just the sound. And then we sort this automatically with an algorithm that tells, okay, this is chow, if the animal is drinking here, eating here, or an artifact if it's a sound but the animal is far away from the food, or Haifa diet if it's nearby this region, or knowing when the animal is actually biting a plastic. So that's another behavior that's very difficult to discriminate from feeding. And after the algorithm do the work, we finally have a human validation, which is very important. So a human actually see all these snippets and correct the position. For example, this one, the tail is touching the chow. So the algorithm is saying is eating chow, right? So the human has to move the folder from here to fix it, and then we build a feeding ethogram, which is human curated, and it's highly precise, very precise, with a very rapid resolution. Here is an example of this ethogram of 6 animals in a fed and a fasted animal, so you can see how they actually eat. So they eat less when they are sated, and they eat more when they are hungry, right? And you can actually build the feeding time in real time and detect precisely all the food intake. All right. What happened with semaglutide, we ask. Can you play the video, please? Here we go. Okay, so it has an audio, but I don't know if you can play it. Well, you cannot hear it, but it has the sound of the bites. So sorry for the Okay, so what you see in this case is very simple. When the animal is sated and you inject saline, the animal eat a lot of the high-fat diet and less of the chow, okay? But once that we put a hungry animal, 18 hours food deprived, and we put semaglutide, what happened is they just eat less, but most of the food that they eat is chow, not high-fat diet. And the surprising thing is that the next day, without the drug, the next day, they also avoid the high-fat diet. So apparently this Ozempic drug is not just inducing a food suppression, it's actually changing the preference of the mice. So they no longer like the high-fat diet. And we discovered this with the crunchometer. Okay, it's a problem with the audio, but it's okay. So what happens if we activate the GABAergic neurons, but now not with optogenetics, but with chemiogenetics? So this drug design, you inject a DRET, you inject a CNO, and the animal will start, we are overactivating these GABAergic neurons, right? So the animal, you can see here how crazy the animal is eating or all this food. Oh, now we have the sound. And the program actually counts how many feeding bouts occurs if there is a high-fat diet. It's so crazy. This is speed, but actually in a normal speed, it's actually very surprising to see. It basically destroys everything. Go ahead. Oh, yeah. This is a sated animal, so basically no bites, like 1-2 bites. This is completely abnormal, like a stress-like feeding behavior, right? So this is what happens when you activate these GABA neurons. You can see in the paper the fine results. But we can also synchronize this the crunchometer with neuronal activity, with electrophysiology, or with microendoscopes, so we can see how the neurons respond. And you cannot do this with these fancy equipment, All right. basically, this is a microendoscope. So here we have different GABA neurons. We can track the trajectory, and here's the activity of all these cells. And this is the alignment with the feeding behavior of this in this session. So here in this session, the animal only eat chow and then lick for sucrose, here in blue, all right? What was surprising was the neurons that respond to food, they don't respond to liquid. So check these 19, 18, and 16 neurons. They don't respond during feeding solid food, but they respond for liquid, during liquid intake. That means that the neurons that respond to liquid food are not the same that the neurons that respond to solid food. So that's why it's so important to measure the food intake of solid food and the microstructure of solid food. So this is another experiment where the animal just ate high-fat diet. You can nicely see how the animal has these 3 bouts and the activation of these neurons that follows. So the GABA neurons actually also respond to solid food, not only to liquid. So in conclusion, the crunchometer is a low-cost open source technology that democratizes feeding analysis, enabling the precise dissection of appetite neural circuits, and it's open to whoever wants to use it. Thank you very much. Yeah. We can. If not, we can share.
Okay. Yes. You need that for the.
Thank you for your presentation. It was very interesting and thrilling. I was wondering if you are planning to study the projection of these neurons, or you have some hypothesis, speculation, where these GABA and glutamatergic neurons are projecting to?
It is already known. So the GABA neurons project to VTA, to GABA neurons and VTA, which disinhibit dopaminergic neurons. So the ones that the GABA neurons in LH are active, they disinhibit the inhibition, so dopamine is released on nucleus accumbens. That's why it's rewarding. And the glutamatergic has the opposite. It activates the GABA, so it decreases dopamine. And it has many connections at different parts of the brain.
But I was talking about your specific study. You can optogenetically activate or inhibit pathway directly. Someone else have done it already, or not?
We are only modulating the somas, but next studies we are studying the fibers and the projections. Yeah. I think that there are so many questions that still we need to answer for the somas, that that going for the circuit is like another 20 years of research. Yeah, but the people have done it. Yeah.
More questions? Yes.
Excuse me, what do you think is the relation between this and the eating disorders?
Oh yeah. For example, for the first neuron that I talk, the AGRP neurons, the hunger neurons, have been related. Another way to inhibit these cells is not only with food, with calories, is by moving, right? So they are being related to binge eating and anorexia nerviosa, right? So somehow these persons with this syndrome learn to inhibit these cells with movement, with exercise. Okay, in an aberrant way. Okay, so instead of eating, they shut down the cells by exercising. And this is particularly present on females. Okay, so there is a paper about that. And for obesity, these neurons are actually, the normal activation is affected, both neurons, the GABA and the AGRP neurons. So the GABA neurons are overactive during obesity, and the AGRP neurons are less able to shut down, to inhibit. So the caloric sensing is kind of affected, of the prediction that this food will have calories is affected during obesity. So that's what is done. Okay.
Another question.
I didn't have time to show that data, but.
I am curious about the experiment with the semaglutide that changes the preference of the food. How long does this effect last?
Yeah, in mice, we're using just a small dose that lasts 18 hours. We know this because we can track the animals for 24 hours. So the suppression of food is just 18 hours, okay? It's not like in humans, like you inject every week, and the half-life is 165 hours or something like that, 7 days. So we are sure that the next day has no drug on the system. But we have other experiments with liquid where we can track the food intake of a liquid high-fat diet for several days. And what we observe is actually very interesting. The day that we inject one single dose of semaglutide, the animal don't drink sugar or lipobenoe, which is a high-fat diet, liquid diet. The next day, they try to eat a little bit of fat, but they apparently don't tolerate it anymore. So the next day, they start drinking less and less and less, and the sugar is up. So they tolerate more the sugar, but less the fat. So there is something special about the fat that these drugs is affecting.
I have a question, if I can.
Sure.
Yeah. Do you know if the circuit is changing in the two-type diabetes?
Probably yes. It's changing in obesity.
Yeah,
so
but it's not the same.
It's the same, right? Like diabetes is just the ultimate state of obesity.
Then you think maybe
it will be worse. It will be even worse.
Okay. Thank you very much.
Thank you.
Are there another questions?
Another question?
No? No?
Thank you.
Okay. Thank you very much. Gracias. Sí. Okay. Then our last talk of this symposium is a video, I think. Yes? It's a video. Yes? Please, this is the Dr. Máximo León Vázquez, President of the Mexican chapter of the International League Against Epilepsy. Dr. León will speak, I think, on neurodegenerative disorders, an important challenge in the region. Please.
Hi, good morning. I'm Dr. Máximo León Vázquez, President of Mexican chapter of the International League Against Epilepsy. It's my pleasure to present today this topic, neurodegenerative disorders, an important challenge in the region. First of all, it's important to talk about the global burden of disease. This study in 2021 reported the 10 conditions with the highest age standardized dailies were, for example, stroke, migraine, epilepsy, and other neurological conditions. The burden of neurological condition is the top-ranked contributor to the global disability adjusted life years or dailies. Global daily counts of neurological disorders increased, for example, by 18% from 1990 to 2001 and affect more than 40% of the global population. The increasing prevalence of the neurological condition is closely tied to the global demographic transition characterized by an expanding elderly population. The impact of neurodegenerative disease extends beyond individual patients, affecting their caregivers. It's an important issue to take account. Alzheimer's disease and other dementias represent a significant and growing proportion of the global burden of the neurodegenerative disease. In fact, the Global Burden Disease in 2021 report indicates that the number of people living with Alzheimer's disease and other dementia worldwide has increased substantially. In fact, as the latest estimates, approximately 21 million people were living with dementia globally in 1990. And nowadays, 56 million people were living with dementia in 2021, according with this report. In this story, a cross-sectional story of 46,000 Chinese adults aged 60 years or older suggested that the old age, female sex, parental history of dementia, rural residents, fewer years of education, being divorced, living alone, for example, is interesting, smoking, hypertension, and other cardiovascular disease, example, diabetes, heart disease, and cerebrovascular disease were risk factors for dementia. Other studies suggested that increased dementia risk was observed in physically inactive individuals who develop cardiometabolic disease. In fact, public health interventions targeting these modifiable risk factors could reduce the incidence and delay the onset of dementia. In this figure, we can see the prevalence of dementia according with the residents. We can see an increasing prevalence in rural areas. It's an interesting fact. According with the Global Disease Report in 2021, the number of people living with Parkinson's disease, for example, has been steadily increasing, largely due to the, I think, aging population and increased life expectancy. In fact, in 1990, approximately 3 million people were considered to have Parkinson's disease, while it was estimated that approximately 11 million in 2021. High-income countries where the average age is relatively high report a greater prevalence of Parkinson's disease. Both in low middle income countries, for example, in our country, are experiencing a rapid increase in Parkinson's disease cases, partly due to demographic change, and are more likely to be exposed to risk factors such as environmental toxins. In fact, for example, pesticides use could an important issue that for this disease. In fact, the increasing prevalence of neurodegenerative disease has exposed significant gaps in the current medical and social support systems. However, only of the increasing global burden, access to the essential medication is not available for a large proportion of patients with a neurodegenerative disease, especially those living in low middle income countries as our nation. These unmet needs are multifaceted and encompass a range of issues that affect both patients and their caregivers. Addressing these gaps is crucial to improving the quality of life, care, and equity for those affected by these conditions. Early diagnosis and intervention is a very important issue. In fact, are critical for managing neurodegenerative disease effectively. However, many individuals do not receive a timely diagnosis because of a lack of awareness, stigma, and limited availability of diagnostic tools and services. In fact, early detection allows for early initiation of treatments which can help manage symptoms and potentially slow the progression of the disease. I think this last point is very important to take account. Other issue, long-term care and community integration is very important. As neurodegenerative disease progresses, many individuals require long-term care, which can be challenging to access and afford. In fact, there is a need for more affordable long-term care options, including home care, assisted living facilities, and nursing homes. Furthermore, there is a growing recognition of the importance of a community integration as the need of age-friendly and dementia-friendly communities. It's very important for the elderly population. According to the research and innovation, despite significant advances in research, there is a lack of effective treatments and cures for many neurodegenerative disorders. Increased investment is necessary in research. Is needed to better understand the underlying mechanisms of this disease, develop new therapeutic approaches, and improve existing treatments. Additionally, there is a need for more translational research to bridge the gap between scientific discoveries and clinical applications. Increasing funding for research into neurodegenerative disease is crucial for discovering new treatments and potentially curative therapies. Investment in basic, translational, and clinical research can lead to breakthroughs in understanding the underlying mechanisms of the disease and developing more effective interventions. Public and private funding sources should be leveraged to support this vital research. Other important topic is policy and advocacy. In fact, effective policy and advocacy are essential for addressing the unmet needs of individuals with neurodegenerative disease. In fact, policies should aim to improve access to care, reduce healthcare disparities, and ensure that the needs of this population. Advocacy efforts should focus on raising awareness, reducing stigma, and promoting the rights and dignity of individuals with neurodegenerative disease and their caregivers. For example, in this strategy, Intersectoral Global Action Plan on Epilepsy and Other Neurological Disorders, there are strategic objectives. In fact, the Intersectoral Global Action Plan on Epilepsy and Other Neurological Disorders has the following strategic objectives. Number one, raise policy prioritization and strengthen governance. Provide effective, timely, and responsive diagnosis, treatment, and care. Implement strategies for promotion and prevention. Foster research and innovation and strengthen information systems. And finally, strengthen the public health approach to epilepsy. I think this is a very interesting plan to fight against epilepsy. In this figure, we can see an awareness campaign against epilepsy. In our chapter, we promote this campaign in 2021. This campaign was focused in rural locations with indigenous people. In this video, we can see the campaign about this topic. Other topic is challenges during and after COVID-19 pandemic. With the widespread to the COVID-19 pandemic, emerging evidence has suggested links between COVID-19 and neurodegenerative disease. More seriously, COVID-19 infection might be considered responsible for the potential causes of symptom worsening, disease progression, and management challenges associated with Parkinson's disease, and dementia. It's very interesting. In addition, individuals with neurodegenerative disease, such as Alzheimer's disease and other dementias, as well as Parkinson's disease, are at increased risk of severe complications from COVID-19. In conclusion, we can talk about for several topics. In fact, the increasing global burden of neurodegenerative disease presents a significant challenge to healthcare systems, societies, and individuals worldwide. In fact, the demographic shift toward an aging population, coupled with the ongoing impacts of the COVID-19 pandemic, has highlighted the urgent need for comprehensive and coordinated efforts to address the unmet medical and social needs. To effectively manage the increased burden, several key improvements in healthcare policy and practice are essential. These, I think, include developing international partnerships for medication distribution, implementing subsidy programs to make treatments more affordable, training more healthcare professionals, and increasing funding for research. Additionally, partnerships, fostering public-private awareness campaigns, and formulating comprehensive care plans that integrate physical, emotional, and social support are crucial steps. Policymakers, healthcare providers, researchers, and communities must work together to create a more inclusive and supportive environment for those affected by neurodegenerative disease. Thank you very much.
Thank you. I want to, we don't have questions because it is not possible to answer them. But thank you very much to assist to this symposium. And right now, we'll begin the second symposium. Then I hope you stay with us. And I don't know, Dr. Sanchez will moderate this symposium. Thank you.
Thank you very much, Dr. Fornaguera. So we continue with our second symposium. Integrative neuroresearch in Latin America, a luxury or a necessity. Neuroscience research in Latin America face enormous challenge of balancing the local needs with global standards. So this symposium aims to talk about whether this is a luxury or an actual necessity, bringing our resources together. So I'm going to first introduce you to Dr. Martín Camarota, who will have our next talk. Dr. Camarota is a neuroscientist and professor at the Brain Institute of the Federal University of Rio Grande do Norte, Brazil, where he leads the Memory Research Laboratory. His work focuses on the neurobiological mechanisms of memory processes such as consolidation, extinction, and reconsolidation. Round of applause for Dr. Camarota.
Hello. So it's working. It's okay there? Yeah? Well, thank you, Laura, for your introduction. Good morning, everyone. So, ¿es mejor con la mano? Okay, now it's better. Good morning, everyone. I'm here to speak about something, about the neuroscience of learning and memory. And why to speak of learning and memory? Because to speak of learning and memory is to speak of the very foundation of our mental life. Every concept that we retain, every habit we develop, and every relationship we form depends on the brain's ability to learn and to remember. When we reflect on personal identity or the construction of culture, we realize that without memory, there would be no continuity, and without learning, obviously no adaptation. Modern neuroscience has made remarkable progress in understanding these two processes. Yet, we are still very far from having an unified theory about learning and memory. What we currently have is an expanding mosaic of discoveries, shedding light on different levels, from molecular changes at individual synapses to distributed neural networks that represent entire or complete experiences. Today I would like to share with you 3 key points. First, a brief but updated overview on how we currently understand learning and memory from a neurobiological perspective. Second, a reflection on emerging challenges in the field. And third, a focused look at the situation of learning and memory in neuroscience in Latin America, exploring our strengths and virtues as well as our limitations and opportunities for short and for long-term development. So, what do we know today? Since the early experiments demonstrating long-term potentiation, we have known that synaptic connections are not static. They are strengthened or weakened by experience. Today, we know and we understand that plasticity, while still a central organizing principle, goes beyond isolated synaptic changes. It includes structural reconfigurations or dendritic spines, adjustments in the presynaptic release of neurotransmitters and neuromodulators, and the action of retrograde messengers like nitric oxide or arachidonic acid. Beyond this now classical understanding, we have discovered that the persistent storage of memory requires the activation of specific enzymes at precise times and brain locations. It also needs the differential expression of genes, epigenetic processes, and changes in large-scale functional connectivity. These findings, all of them, remind us that memory is not a single mechanism, but a constellation of interdependent processes that operate across multiple levels. This brings us to the concept of memory reconsolidation, which has gained substantial relevance over the past 25 years. Reconsolidation refers to the process by which previously consolidated memories become temporarily destabilized when recalled in the presence of new significant information. This process allows those memories to be updated before restabilizing. In essence, the empirical evidence or demonstration for reconsolidation not only has revitalized a classical concept for experimental psychology, but also confirmed something intuitively familiar. Memories are not fixed, but every time we remember, we reopen the possibility of changing what we recall. This deceptively simple idea has profoundly transformed our understanding of how memory works, opening up a new line of inquiry with far-reaching theoretical and practical implications. Theoretically, the existence of a reconsolidation process raises a provocative question. If every time we recall a memory, this memory can be altered, could the most accurate memories be the one we have never accessed again and don't even know we have? From a practical point of view, reconsolidation has become lately a key focus in developing treatments aimed at modifying traumatic memories, such as those involving post-traumatic stress disorder. The plasticity of memory suggests that we are not only capable of forming memories, but also of transforming them in target ways, opening therapeutic possibilities for conditions that are rooted in our emotional past. Despite these advances, many studies still study our brain structures or different brain structures in isolation. In this way, the hippocampus and the different rhinal cortices remain central to declarative memory research, while the prefrontal cortex is essential for working memory and systemic consolidation, and the different nuclei of the amygdala are key to emotional processing, while the insula plays a major role in aversive memories. Even the cerebellum, traditionally associated with motor control, is not recognized as a fine modulator of cognitive learning. This distributed and dynamic view allows us to affirm that memories are not stored in a single location, but rather in patterns of electrical activity involving multiple circuits. Still, memories are not just patterns of activity. The molecular identity of memory traces remains an active area of research. For example, we now know that non-coding RNAs and chromatin remodeling contribute to the stabilization of changes that allow experience-derived information to persist over time. This fascinating field opens the door to convergence between learning neurobiology, molecular biology, and epigenetics. The technological leap of the past 2 decades and a half has been decisive. Tools such as optogenetics, chemogenetics, high-resolution functional imaging, active neuronal tagging, and transsynaptic tracing techniques now allow us to causally manipulate memory-related circuits. Thanks to these new tools, the concept of engram, originally proposed over a century ago by a German zoologist called Richard Semon, has made a powerful comeback. Today, we understand the idea of this engram as a set of neurons and their structural and functional interconnections, which form the basis of a memory. We can now identify these neurons, we can now activate these neurons, we can now silence these neurons, and even modify the content of memories. This new conceptual framework has transformed the relationship between learning neurobiology and artificial intelligence. This is not longer science fiction. Artificial neural networks inspired by principles of plasticity are now helping us to generate testable hypotheses about how we learn and how we forget. Conversely, computational models provide neuroscience with new ways to conceptualize complex phenomena, such as the emergence of properties in systems composed of billions of neurons and glial cells. This reciprocal interaction between artificial intelligence and neuroscience is one of today's most promising and fertile dialogues. However, the major challenge lies in experimentally validating our hypothesis. This requires integrating levels of analysis from the molecular to the behavioral, while accounting for individual variability. Factors such as stress, age, sex, and cultural context all shape not only how we learn and remember, but also what we how can we change these memories. This highlights or stress the need for standardized methodologies that ensure replicability and ethical rigor. It also compels us to consider the limits of memory manipulation, particularly as findings, such as those from reconsolidation studies, begin to influence therapeutic protocols and experimental technologies. This not only offers new clinical hope, but also raises important ethical questions about memory alterations and personal identity. This is especially critical in aging and trauma-affected populations, where memory disorders are on the rise. And in the world in our actual world, where distinguishing between true and false, or trivial and meaningful, have become increasingly urgent. Conditions like Alzheimer's, other types of dementia, post-traumatic amnesia, are pushing research towards therapies based on brain stimulation, precision pharmacology, and cognitive rehabilitation. For instance, early steps have already been taken toward interfaces capable of recording and stimulating memory patterns. Though still experimental, such technologies raise both exciting prospects and serious ethical dilemmas. Moreover, these insights also call for educational environments that are inclusive, effective, and biologically informed, respecting rhythms of sleep, attention, and development. This represents an area of direct transfer from basic research to public policy, and will only be effective if based on technological convergence. Undoubtedly, the neuroscience of the future will need to be interdisciplinary by necessity, with biology, physics, engineering, artificial intelligence, and philosophy working together to understand a phenomenon as complex as memory. So, to speak of neurobiological neurobiological perspective of memory without addressing the reality of our region would be incomplete. Latin America has made notable contributions to neuroscience research. Indeed, research centers here in Mexico, in Chile, in Argentina, in Brazil, in Uruguay, and Colombia have produced internationally recognized science across fields like electrophysiology, neuroimaging, and neurocognition. Scientifically, our region has grown significantly in the past 2 decades. Although it still represents a moderate portion of global output, there are centers of excellence and highly active collaborative networks. These efforts show that the neurobiology of learning and memory in particular is not only a lab-based discipline, it is also about classrooms, communities, and public policy. We have here in Latin America clear strength, creativity, resilience in the face of limited resources, and strong academic training traditions in many public universities. But we also face significant structural challenges. Low and unstable funding for science and technology, inadequate infrastructure, and unequal access to advanced training and technology. Ethical frameworks for regulating neurotechnology are also often underdeveloped, raising concerns about protecting fundamental rights like mental privacy, personal identity, and cognition freedom. We also remain heavily dependent on international partnerships and imported equipment, creating a difficult cycle. Talented researchers, but limited capacity to sustain long-term projects or retain top human capital. To break this cycle, we need to build and strengthen regional collaboration networks, sharing equipment and data, supporting students' mobility and lab exchanges, and promote open science and free access to publication and datasets. In the long term, we should aim to establish state-level policies ensuring stable research funding beyond electoral cycles. We also should aim to create regional neuroscience centers of excellence which share high-tech resources, incentivize the return of researchers trained abroad through competitive programs. This last point may be the most critical. Because it is not enough to train researchers. We must create ecosystems that retain and empower them. This means designing regional PhD and postdoctoral programs with co-mentorships across countries, funding consortia for early career researchers, and integrating neuroscience with broader discussions in education, public health, and social policies. So neuroscientists can take active roles in transforming the region's realities. Learning and memory are not just research topics. They are the essence of human experience. To understand them is to understand ourselves. Today, neuroscience offers extraordinary insights. The neurobiology of learning and memory, in particular, is undergoing a conceptual and technological revolution. We are shifting from studying isolated parts to understanding dynamic, plastic, adaptive systems able to integrate individual experience with biological foundations. Latin America despite its limitation, it's not excluded from this process. It has generated original knowledge, built collaborative networks, and began translating findings into educational and clinical practice. Much work remains to be done, but the path is already open. We have both the opportunity and the responsibility to think about how this research fits within our own social and political realities. The future of the neurobiology of learning and memory will certainly be global, but it can have a distinctly Latin American accent if we sustain scientific excellence, foster collaboration, and train new generations of researchers who not only study the brain, but dream of transforming the world around them. Because ultimately, to study how we learn and how we remember is not only a scientific pursuit, it is a way to understand who we are, how we grow, and how we might build a more just, informed, and human future. Thank you very much.
We have time for questions. Questions in the room? So I have a question regarding the Latin American accent. What is it that we can provide to the region to make sure memory research goes beyond where it is right now?
In the neurobiology of learning and memory field, you mean? Well, what we need to do, I think, is to have a more integrated perspective to the field. We must share resources. It's not a diaspora, as we were talking the other day, but we have to put all our efforts together. We don't do that in the field. We have several labs that are usually working on the same things, but we are doing that independently. This has a clear output. In this way, we are never gonna be able to, in a practical way, to publish in high impact journals. To do that, we, in Latin America, we need to research our resources. In the neurobiology of learning and memory field, it is extremely competitive, and it is how can I say it? It's a very small field. So we certainly need to share resources.
Hi, Martin. Nice talk. I have two questions, one scientific and one for policy. So it's very nice, this update that you talk about memories, that they jump, they move, they are dynamic. And it has been thought that they are mainly in the synapse. But do you know what they know now for the soma? Also the soma is involved. And you mentioned something interesting that I might not be agree completely, like the patterns in spatial-temporal patterns of activity don't encode the memories, right? You're right when you say if we put electrodes and we try to measure the memory trace just in the spiking of the cells, we may get lost. We might never find these memory traces, right? But after these spikes are happening, intracellular components in the cells last for hours. So maybe it's what is happening inside the cell that we don't have access, but that are triggered by the spikes, right? So maybe the memory is happening inside the cells in intracellular processes. That's a scientific question. And the second question is...
I have to remember the
okay,
just
it's
okay, but
okay.
Go on.
The second question is, do you have a plan or an idea of how to convince politicians to sustain funding? Because these waves of fundings are really killing any scientific field. right? We are just happening one here in the last 6 years. It has been like up and down, more down than up. Do you have an idea how to convince politicians? And it's also right now happening in the US. So is there a way to do it?
So the first one, your first question is easy.
Okay.
No, it's easier to respond than the second one. So to the first one, we have to take into account that when we talk about the engram, when we talk about dynamic processes and so on, this is something that we usually forget. When we do all that, we are not measuring memory. We never measure memory. We only see biochemically, electrophysiologically, or whatever, a memory when the animal or where we are using that memory, when we are expressing a behavior. So actually, all those changes, the engram, the dynamic, and whatever, is not memory. It's memory in motion. So what we are measuring, what we are registering, all those changes are memory in motion, are memories being reactivated and used. So what I mean when I say dynamic changes is that now we realize that. And we can develop the proper control groups or the proper control experiments to separate memory as an engram that is stored in some place or in some form in the brain, and memory in motion. And this has taken us to the idea that maybe memory is not a thing. Memory is not a representation. Memory is the ability the brain has to build up and use representations that change all the time because actually they were never there. So the metaphor of a film or of a photograph, obviously it was always wrong, but now it seems that it's wronger than never. You know what I mean? It's not a thing, it's not a representation, it's the capacity we have.
It's just to give meaning to experience, right? It's not a photography.
But this has been always so, in that way. I don't know how to put it in words in so short a time, but it's a completely and revolutionary framework in the neurobiology of learning and memory field. The idea that memory is not a representation of any kind. It's not a psychological representation, it's not a molecular representation, it's not an electrophysiological representation. No. It's the ability, the changing ability, the ever-changing ability to construct, to build a representation. So what actually changes is not the representation, because the representation changes every time. If you want to imagine your mom with green hair, you can do it. Yeah. And this is a representation. This is a sort of false memory. Yeah, you know what I mean? So what changes is your capacity, or the brain's capacity, to build, to express those representations. This is the change in paradigm. I don't know if I understand. And the second one, well, the second one is extremely difficult. In the neurobiology of learning and memory field, which is such a small, powerful, but small field, is a sort of boutique field, if you want. What I think that we have to do is to build up small consortium of 15, 20 researchers that make meetings like this one, small ones, where we can discuss about what we are doing, but really discuss and show us the actual experiments, the actual data. And I know it's a dream and it's impossible, or whatever, because we have to admit that we compete with each other for the same resources. So But actually, and this is the point, we compete for researchers from abroad. So if we amalgamate, if we forget about our egos, and we can present a strong proposal, maybe we can do a better science, whatever that means. Thank you very much.
Hay un comentario en línea. Hay un comentario en línea. ¿Sí? ¿Cuál es?
Hello, Esper.
Okay. Good morning, everyone. I'm so happy to be here just to say some words to Martin because are you listening?
Esper.
Yes. Are you listening?
Long time no see. How are you?
I'm okay. Just to say that you that you are reminding me my past, what is not really good for me, reminding my past, but to say that just to comment your reflections about how difficult it is to do science in Latin America. I think that your reflection was very good to the young generation because it's not easy and sometimes we feel ourselves that we are just repeating things, doing the things again and again because it's so difficult to have the right support. So this is just my comment by now. Thank you.
Thank you, Esper. Yeah, sure. I didn't catch what you say because we have a lot of reverberation here. But maybe if someone want to make a comment?
Don't worry. Go ahead with the lectures. Okay.
Nice to listen to your voice again. Bye-bye. I see you soon.
Now we're going to continue with our symposium. I just want to make sure we have the connection because our next guest is presenting live via Zoom. I'm going to introduce him first. Dr. Pedro Valdez Sosa is a Cuban neuroscientist and co-founder of the Cuban Neuroscience Center, where he currently serves as Vice Director for Research. He is internationally recognized for his pioneering contributions to brain mapping, neuroimaging, and neuroinformatics in Latin America. Let's give a little bit of time for the connection to come through. Here you go. Good morning, Dr. Valdez. Go ahead.
Oh, good night. Good night because I'm in China.
Good night.
So basically, let's see. First, I want to see if you can see my screen. Can you see my screen? Can you see my screen?
We don't just yet.
No.
Are you sharing the screen?
I'm sharing the screen. Can you see it?
It's coming.
I'll try again. Just a minute.
One quick second. We're checking the connection here to make sure we can see your screen.
Can you see it now?
No, we can see you.
You can see me, but not my screen. Okay, that's not good. I'll share again. And now?
Not yet. I'm going to ask people from our end. Probably we are sharing our screen. Does that allow him to share his own screen?
It's okay now.
Is it good now? Is it good now?
Yeah, you have to. Yes, but you have to do it bigger. You know, it is your screen, not your show.
Got it. And now?
Okay, perfect.
Hi, it's Pedro. Okay, guys, can you hear me?
Yes.
Are we ready to go?
Yes.
Should I start? I just need to know.
Go ahead.
Should I start? Okay. Okay, first, greetings from China. That's where I'm at this moment. I am now in charge of a laboratory here in China, which is the Cuba-China Laboratory for Neurotechnology. We created it 10 years ago. Now it's become a national level laboratory dedicated to integrating research not only between Cuba and China, but also with the rest of Latin America, and in fact with all the countries of the Belt and Road and BRICS initiative. At this moment, we are at a high-level meeting with the Cuban Vice Premier and the leaders of the Chinese National Commission of Reform and Development, studying how to integrate more fully with Latin America. So that's why I'm here in China most of the time, and I'm also now speaking from China. So my presentation is going to be how to bridge scales across different types of images and other types of information. And I'm going to be mentioning the Big Brain Project, which is a joint project between the Helmholtz Institute and the Montreal Neurological Institute, where I also hold a position. So I think that they have connected the. Let me just get rid of the race, okay? So from our laboratory, we have created here with Canada something called the Global Brain Consortium. The Global Brain Consortium has now 35 countries, many from Latin America, and the idea is to share open source data and also to share procedures for analyzing brain images across the world. For this, we are using a shared platform of IT. And here you see the members of the steering committee. On the one hand, there's Alan Evans and myself, who co-direct the Global Brain Consortium. And I'm just going to mention Patrick Nonmans, who was the scientific director of the European Brain Project, Paolo Rossini from the International Federation of Clinical Neurophysiology, and Michael Milham. There's some more, but there's not time to present them all. So basically, the problem we have is that we can create narrow images from all types of activity that is occurring in the brain. Let's take, for example, transmission of signals from one neuron to the other. We have postsynaptic activity, we have neurotransmitters, that have spikes, and this produces changes in glucose and energy consumption, and the changes in blood vessel size and volume, and the blood flow, and so forth. Now, each of these produces a type, can be used or leveraged to produce a type of narrow image. And up to now, we've been looking at them separately. The first target of the Cuban Center for Neuroscience and of the Global Brain Consortium has been electrophysiology. Here I summarize some of the techniques we've developed over the years. This is a Cuban system for recording EEG, which we developed. We started developing these systems in the 70s. Here we see the placement of electrodes, EEG activity, electroencephalography. We single out activity from one of the electrodes, we can construct what is known as the cross spectrum, just the covariance matrix of the Fourier coefficients. If we look at the variances, we then have the spectrum. Here we see the peak in the alpha rhythm. In the power spectrum, we can plot it on the surface of the head. If we look at the elements outside of the diagonal, the covariances of the Fourier coefficient, we can study functional connectivity. It's a bad name because there's nothing functional about the covariance between electrodes in the frequency domain. And this is known, this is the cross spectrum. Here we see a map of this. Actually, what we're interested in our work is to consider a biophysical model where we look at the sources inside the cortex, and then we see how this is projected towards the electrodes. And this is the forward model. And with that, we created in the late 90s, here's an image from 1996, where the origin of the alpha rhythm is more, or the occipital area. We can then, and this is more recent, move and look at now the cross spectrum, or the directed connectivity inside the brain at the cortical level, estimated using source analysis methods. And this is a bit more actually related to something called functional connectivity, because it's not the reflection of indirect sources that are just like shadows on the surface of the head. Most recently, we have worked in the Global Brain Consortium to create a network. You see the countries that contributed data to create lifespan norms to be used in clinical settings. And this is the pipeline. I won't go into details. Basically, each country and each site, so there's 16 sites and 9 countries. This is about 2,000 subjects, or participants, which were completely normal. And you see the age distribution on the bottom left. Now, the point here is that either they share the EEG or they share the crossbreed. It went through a pipeline where we did automatic detection of outliers. We could even find places that were putting the electrodes on in the incorrect order. From there, we can create what we call developmental surfaces of all the parameters. That is to say, for a given value of a parameter over all frequencies, from age 5 to roughly 100. With that, we can then do that for the mean of the parameters and the standard deviation and create Z-transform maps of all the parameters. And on the extreme right, you see the distribution for all batches in all countries. And what we found is that it was necessary to harmonize data, just as in bioinformatics. And we'll see the importance of this in a minute. All of this software is publicly available and downloadable. All the data is publicly available and downloadable. And there's been a significant contribution from Latin America. Now, 2 examples of the use of this. One is the study of malnutrition and its effect on the brain. And this is a long-going study in Barbados from the late 60s, early 70s, in which there was malnutrition, severe malnutrition from children. You can see here in the image on the left, one child with this type of malnutrition, protein energy malnutrition, and the normal controls, which were from the same neighborhood, same characteristics, and these have been followed up for more than 50 years with the cooperation of these people and of the authorities in Barbados. This study that's being done together with Harvard allowed us to gather in the in 2000 in 1998, I'm sorry, when the children were in school age, and later, 2018, when the people that were participating were not 5 or 11 in school age, but were rather 40 to 50. This allowed us to study the EEG at these 2 occasions. I have to say that this is a very rich study. There's epigenetic changes that are transmitted to the 2nd generation. There's attention deficit disorder. Everything is very documented. And in the people, when they are already in the 40s and 50s, there's more mild cognitive impairment in the people that had the protein ownership malnutrition. So using these norms that I described, we took a subsample of students to use to correct for the batch effects. And what you have on the right is the receiver operator characteristic of different procedures using improved statistical methods and harmonization. I only have to say that if you look here on the right for the area under the ROC curve, harmonization and correct statistical procedures allow you to go from 0.83 to 0.95 of discrimination at school age. And this is valid also for older age of the group that had malnutrition and the one that did. A second study, which I want to mention, was done with Francisco Lopera, our departed friend, and this was with the families that were detected by his group in Medellin that had the presentation-1 mutation. The Cubans, we were able to take it. This was a very old model system in Colombia, and we started physiological studies there that had later been extended by the group there and by other collaborations. But I just want to mention that this was one of the first studies where we found between controllers of the family, people, the family, members of the family, that is, that did not have the mutation, those that did have the mutation, and different stages of this, we were able to find very significant differences and classify the subjects in a perfect accuracy using the source analysis I was telling you about. What you see on the right is this, the areas that our methods at that time allowed us to discriminate between these groups. This is a very early example of molecular imaging using EEG. Okay, these are the two examples. Now, the point is the following. We're only looking at EEG. As I say, we're in some cases at spikes, in some cases at postsynaptic potentials. How can we try to bridge the different scales? And here is where it is very important to mention that now we can leverage for neuroimaging connections with neurotransmitters, with behavior, with gene transcription. This is a recent paper in which our group participated with our Chinese colleague. The head of the group is Peng Zhu, and here, very briefly, because that could be the center of the rest I'm going to talk here, what was found is how there was variability between functional connections as measured by fMRI and structural connections as measured by diffusion laser imaging, and this allowed to create signatures of this structural functional variability, which then were compared temporal variability of the structural functional connectivity with behavioral traits using multivariate methods, with imaging transcriptomics. There was then the possibility of looking, for example, and this is here, the effect of different neurotransmitters on these characteristics. So what we're seeing is attempts to bridge from the whole brain fMRI activity and then try to link it to more molecular features. And this is a characteristic of research nowadays. This has become much easier, and that is the title of the talk, under the Big Brain framework. This was a project, a paper in Science, between Catherine Emmons and Alan Evans. And here you see how they were able to use a macrotome to slice the brain. This was just one hemisphere that has been now replicated in other participants, let's say not participants, other measurements, and then in monkey. And here what you're seeing on the moving image in the movie is how, by computer analysis, you can reconstruct cuts and tears. And what was created was an atlas. And around this atlas, we've been building a number of in this common space. So this would be the successor to the Montreal Neurological Institute Atlas. This is the Big Brain Atlas. And there's now plenty of software to combine individual images to this or this type of big brain to other images. What has been done? Now, what's been done is to try to produce in this project. This is the page of the Big Brain project. You can find more details there. What we're trying to do now is in an easier way to compare different types of images. And I want to give a shout out to this Nature Methods paper on what's known as narrow maps. And this was developed by Marcelo. This is the group of Bratislav Mishik, with extraordinary work by Justine F. And basically what you see here is that into the big brain space, they've pulled together information not only from MRI or functional MRI or MEG, but also from PET data, from data from the Allen Institute, and it's all in a common space with statistical methods. Briefly, for those who don't know about it, they might get interested. The simplest one, which is not the most accurate, is to represent everything in the cortical sphere, and then to create a null hypothesis of similarity over space by rotating randomly the sphere. The methods have improved. But basically, this has posed challenges on how to compare with small and large samples the different types of data. Our task within this at the Cuba China laboratory has been to fit electrophysiology into this. On the left, you see the source activity on the big brain. On the right, whole brain modeling, using neural modeling of the brain, also in the big brain space. This is now being called digital twins of the brain. The idea is to try to make these whole brain models as specific as possible, and we're now trying to build in all the information available in neural maps and other sources. I want to show how we can use this, and the example I'm going to take is a very interesting characteristic of the EEG spectrum, and it is that for many years it's been observed that in addition to the peaks produced by rhythmic activity, for example, we can see there in the curve at the bottom how we have an alpha peak at 10 hertz. There's this activity that decreases. At the time, Roberto Pascual, who's my PhD student, we worked on this, and we call this process that goes down. It's now called 1 over F without any evidence. It's also called aperiodic. We call it the C process. And then we called the alpha activity the alpha process. And we went on to parameterize this with functions. This has become very popular now. There's different names for it. This is old work. This is from 1987, 1988. Become popular now. And people know other, what's been done in the West. Okay, so we've been looking here at the following things. First, are these processes the same from the point of view of statistical process, stochastic process? So for this, we use the database of 1,500 subjects. This is work that's been carried out by one of my PhD students, and we're using the EEG data, but we're also using, as part of the Big Brain project, the atlas that was carried out by Birgit Rauscher. It's been collected, and John Gottman. And this is data from a subject that we're going to undergo epilepsy surgery. They selected the channels that were considered normal. There's more than 1,700 channels from approximately 100 subjects. And what we did was to analyze this activity that I'm saying, this aperiodic or psi process that decreases with frequency, and the rhythmic activity, and to see what characteristics it had. In a nutshell, because I don't have much time, both for scalp and for the intracranial electrodes, we found that the process that decreases aperiodically, and it doesn't have any rhythm, is linear and Gaussian, and the process that is rhythmic is actually a nonlinear resonance. And what that means is it has a peak in the spectrum, but it also has a peak in the generalization of the spectrum to higher order, which is the bispectrum, which measures nonlinear phase coupling between different frequencies. So between the alpha rhythm and its harmonics, you get a very strong harmonic relation, which is present in the occipital area electrodes, but it is even stronger in the somatosensory area, motor area, because that's where the mu rhythm is. And what this reflects is asymmetry. And here we show you some nonlinear dynamical methods that we're using. Will be later used, and it's not the subject here, for the detection of retro elements in epilepsy. And you see that when you have activity that's symmetric, you don't have spike coherence, but you do have spectral peaks. And on the right, you see that we have asymmetrical activity, and this is a dynamical reconstruction we're doing. You do get not only more peaks, which are harmonics of each other, but they are linked harmonics. The other problem that we went into is to try to model this and find this in subject. So we created a model based on reduced thalamocortical units that then we connected through the Human Connectome Project connectivity matrix, and we went on to include delays that were measured in also patients that were going to undergo epilepsy surgery. This is the LATBrain project. So we built into the method for estimating the sources of these activities data from the population that were obtained and who's connected to who, and how much does these delays, how much the activity delays from going from one place to the other. This was then adjusted for each individual, and we fit this for more than 1,500 subjects from age 5 to around age 98. And basically, across all ages, here you see the age progression of the activity in different decades. You see here that the alpha activity is very peripheral occipital. You have some activity, I said, related to the somatosensory area, motor cortex. There you see the numbers. The other process, which is the 1/F, it's more widespread. This concerns what we observed many decades ago. But interestingly here, we can now measure effective connectivity, not functional connectivity. So we can look at which which thalamocortical units that we've modeled are connected to others, and what we see is that it is concentrated specifically in parietal occipital region, and that it is, if you see the emergence over age, you see that there's a characteristic in which it increases, then decreases, and it becomes very clear in later ages that there's two foci, one for the occipital region, that would be the alpha rhythm, another for the mu rhythm, and we don't think that on our recordings we can find tau with the signal to noise ratio yet. The other activity, which is the sight process, is more widespread. It also has an evolution over age. And now that we've characterized this, well, how does this connect to what I was saying before? How do we use neural maps to try to study the connection to basic things? And here we have an hypothesis. This comes from prior work we did studying the human brain mapping database, in which we correlated the activity of how fast the alpha rhythm is, what is the rhythm, with several biophysical hypotheses. The one that was very interesting is that we found that the fractional anisotropy of diffusion-weighted imaging in certain tracks, that is to say, how organized these tracks were, was what related to the alpha rhythm. So we thought that the speed of the alpha rhythm is very much related to myelination. And what we did was then, and here you see this curve, which is very similar to what we find in all the curves that we look at with EEG. And basically, this is a paper on the cycles of myelination. This curve is obtained from precisely what I was saying, T1 divided by T2 in the Human Connectome Project. That's the basic data for this paper, supplemented by other things. So what we did was to look at the delays, the general delays for the whole process in the brain, a general delay factor. And we found that for these 1,500 subjects, the delays diminish with age it reaches, let's say, a peak, and then in older age, the delays start to increase again. Now, what we did was then to convert these delays into a proxy using biophysical models that are well established of myelination. And here we're superimposing the curve I just showed you from that review paper. It comes from T1 divided T2 with our data. So this is our data, this is the one. Now, what did we find? We found that the overlap is considerable. When we did statistical testing using narrow maps, we found that there was no difference between them. So in a certain sense, this strengthens the hypothesis, and now we're checking for neurotransmitters for the different rhythms. And this is just an example of the type of answer that we can do with multimodal integration. Before ending, I want to say that all this work has been possible thanks to collaboration with Latin American countries, but also with China and other countries, and particularly Canada. We believe that one answer to the situation that now exists, you know that Cuba has been under enormous pressure and more now than ever. And what our strategy, that's why we have a lab in China, is to diversify our partners and to look for alliances. And I have to say that together with the Chinese, the Cubans have looked into other alliances. We've been in strong discussions with the countries of BRICS. In fact, we've had several neuroscience BRICS meetings. And we think that a proposal, there could be a BRICS initiative on neuroscience. We don't have to look to the north of our countries only to look for funding. In the second place, there's an initiative called the Belt and Road Initiative. And just a few months ago, we discussed there with 85 countries the possibilities of collaboration. And I think the world has to become diversified in terms of where the funding comes from. There have to be different alliances. None should be preferential. But what we do say from Cuba, from the Cuban side, is that we hope to work with all the people possible to join in any of the collaborations that we're allowed to, because there's certain collaborations which Cuba is being blocked, and that we hope to put all the things that I've said as just one part of what I see is becoming an initiative to have a real community in the neurosciences in Latin America. Thank you very much. Sorry for the quality of the presentation, but it's from the other side of the globe. That's it.
Thank you so much, Dr. Pedro Valdez Sosa. Now we have some room for questions.
Do you hear me? This is Luisa Rocha. Nice to see you, and thanks a lot for your presentation. I think Big Brain Project, it's an excellent project to integrate all the information, the electrographic information of the human brain activity, both in basal conditions, normal conditions, and pathological conditions. However, as far as I know, it is very important to know how to get the information, how to integrate, how to analyze. I believe in the future, it could be an excellent opportunity to organize a kind of course or a project in order to know how to participate in this very important project.
Excellent initiative. Do you hear me?
Yes.
Okay, it's an excellent initiative. That's precisely why we created the Global Brain Consortium, which is very connected to the Big Brain Project. We're very happy to do this, and we're happy also not only to participate in online courses, but to participate in personal, organized locally courses, also to accept. There's a characteristic. While in the US, Trump is destroying science, China is building up science, and we're participating with them in this buildup of science, even if we have economic difficulties. Because science is the future. If without science, we will not be able to address the problems that we have as human beings. So I think that we should see how to raise consciousness in, and that's something that's happening. I mean, we presented to the 85 countries the idea that we have to worry about population brain health. That the governments, I think we should say that this is not just doing research to produce abstract knowledge. This is to solve the basic problems which is affecting aging population. And I think that what we have to do, as was said before, is to integrate, not compete, but complement. Because that's essential, and to not only to be worried about the specific research, but about the research strategies that we should do that will enhance our results, but will also allow us to translate results into society. And I think we have to convince all our governments that we have to put more funding into this, but this will require that we interact ever more, and you have us at, I mean, we are at the, let's say, you can ask us for what you want, and we're willing to do it, okay? We're willing to do it to the extent that we have possibilities.
Thank you, Pedro. And I believe this presentation shows that Latin America has a very good opportunities to collaborate with other countries in Asia.
You're right.
Thank you so much. Do we have more questions for Dr. Pedro Valdez Sosa? Okay, let's give a round of applause to Dr. Pedro Valdez. Thank you so much for your presentation.
There is one question.
Oh, there's a question.
Is one question, the Maria
Marita Castello. Go
ahead.
Marita.
Hello, thank you for the opportunity. Hola, Pedro. Nice to see you again. Yeah, it's a great pleasure to see you in this meeting. You know, very nice your presentation, and I was wondering if when you make this multidimensional analysis, complementing EEG with other imaginology studies, do you use tractography also?
Yes, of course. I mean, that's standard. The figure I showed, there's a figure I showed, that's the mesoscopic brain model. But this is old stuff. I'll send you the reference. There's a 2009 paper where we looked at every voxel, and with tractography, what's the connectivity matrix? And then yes, we do. We build in the using diffusion imaging. That's the anatomical connectivity.
Okay.
And in fact, we've continued doing this. We have a more recent work where we're looking at variable, because people model the brain using the tracks by tractography, and then they put a single conduction velocity. And we know that tracks have diverse sizes of axons. Some are more myelinated, some are less myelinated. Our more recent work puts in this realistic. So what we're working now is to improve this anatomical connectivity to be more realistic. But yes, of course we do. We use it. I'm sorry if I didn't mention.
Okay. Thank you so much.
Thank you so much. So now we move forward with our second symposium. This is the last talk. It's by Dr. Daniel San Juan Horta. Dr. Daniel San Juan Horta is a neurologist and a researcher at the National Institute of Neurology and Neurosurgery in Ciudad de Mexico. His research and clinical work center on neuromodulation and the treatment of epilepsy. I think we have a virtual presentation. It's via video. And here we go.
Hello everyone, my name is Daniel San Juan. I am neurologist, clinical neurophysiologist, and epileptologist. from Mexico City, and for me it's an honor to stay with you today to speak about the neurodiversity, a new way to evaluate mental health in Latin America. So to start this lecture, I don't have any conflict of interest, and this is the agenda for today related with the concepts, some basis of epidemiology, neurobiology, diagnosis, and the most relevant, the interventions. The conventional medical paradigms of this condition is derived from the medical model of disability that's considered autism as a disability. And disability is defined as any restriction or lack of ability to perform an activity in a manner or within the range considered normal for human beings. That includes the existence of a typical level of ability, and in this situation, autism and other disorders are considered pathologies. This approach is trying to identify deficits or symptoms or signs that affect the functionality of the patient. And now we consider there is a broad spectrum of the neurodivergence that include the increase of risk of specific advantage or a specific situation that can be improved with the repetition, or patients that have specific disorders. The goals and intervention in these medical paradigms is reduce the core symptoms that the social communication deficit, repetitive behaviors, decrease the comorbidities associated with the disorders or the interrelations between them. And usually the clinical diagnosis is based in standard criteria from the DSM-5 version and other codes. And the intervention are based on the behavioral therapy, speech and sensory therapies, social skill training, and usually include pharmacological treatments for the symptoms. And you know that there are a lot of the symptoms related with specific situation or condition like dyslexia, dyspraxia, Tourette syndrome, or other deficits like attentional deficit with hyperactivity disorder. But what are the barriers or limitation with this spectrum of the conditions? You can see in the graph the repetitive behavior, communication clarity, the attention and concentration, or other like patterns related with repetitive and very strict patterns of working. And the other side is the issue related with the functional executions of the functions that include the organization skills, prioritization of tasks, or the time management, shift from task to task. This situation emphasizes the deficit over the strength and can promote in some situations the normalization over acceptation and can neglect the autistic person's subjective experience. So the new concept is to consider the autism and other conditions like a variation and no disorder in human cognition and behavior, and try to include this social acceptance and inclusions of several individuals with this kind of the skills that include promote the autonomy in the individuals, respect of communication styles, be informed about the knowledge of the neurodivergent, take in especially in education or jobs, the strength approach, or also include this personalized treatment according to the needs of each subject. So there are a lot of intervention that is possible to apply not only for the medical community, that include the most common settings for one of these subjects that include the schools, jobs, even some common recreational areas for these individuals. You can see in this slide one of these examples about understanding the neurodiversity in the workplace, how is management to increase the productivity of the company and promote a more comprehensive inclusion of the workers. The idea is to promote the self-determination and improve the quality of life of each individual, include the environmental adjustment, and include this kind of the communication, sensory skills or strategies and routines. The collaborative intervention focuses in getting a real impact in the person and the family, and the inclusion programs to try to get new abilities that include the standard schools or special programs. And of course, the human rights perspective to decrease the discrimination, at least for this variety of conditions. This sometimes is difficult because that include the participation of this population in research and policy making. But still, one of the challenges in work is that we have a limited evidence of the long-term effectivity of this program designed strictly under this approach. There are, of course, challenges to implement, not only for developed countries, including Latin America, that include adapt significant systemic change that sometimes is not feasible in all settings. That you can imagine some of these situations when is needed specific strengths of the person to management, for example, an airplane, or tension between the support and acceptance, and the risk of overgeneralization, because not all the patient or subject with autism have this perspective of the neurodiversity, especially those with high support that need more medical attention. And we know that at least in Latin America, Mexico, and in other countries, the prevalence of the neurodivergency is very similar. There are two numbers that is very common to listen is about the global prevalence of autism, around the 1 to 2%, and the neurodivergence in total 10 to 50%. And the more complex situation is when is included the intellectual disability, and this is the global prevalence worldwide. 1 in 132 people is affected. Interesting, is more common to get the diagnosis in males, and that's included now in a bias for the diagnosis. And the global burden for autism spectrum disorder, as you can see in this graph, and the disability adjusted life years is 3.32 millions. That is very high, and unfortunately is affected more in the childhood and adolescence, reflecting the early onset and the constant disability. They are not only for autism, for other mental health conditions, genetic factors, perinatal factors that you can see in the graph, how is the weighted. Some are very clear, like neonate hypoxia, or use some drugs like evaporate the maternal age more than 40 years. They include the paternal age, 1 decade after paternal birth, another factor related with the increase of the risk, and the environmental and social factor also are relevant. We have some of these problems in the region because that include a high rate of prenatal exposure to pesticides, the summer bird. That means it's possible that the regions is not so cold like in Nordic countries, or this related with the perinatal care, the premature ruptures of membrane, that is related with the habits of the population and the medical health system to provide this assisted vaginal delivery or C-section or the care of the mother during the pregnancy. And there are this interaction with these factors, but at least for the moment, there are 6 theories about the cause of this situation that include the disruption in brain development and structure, white matter and connectivity, social brain network disruption, default mode network abnormalities, sensory processing with over or under activation, and of course, a neurochemical disbalance in the excitability and some risk like hyperserotoninemia in some individuals with autism. I don't have time to explain these experiments and that can prove some of these deficits. And the standard diagnosis is basic in for the medical community using these statistical manuals that include, at least for autism, the persistent deficit in social communication and interaction, and restrict repetitive patterns of behavior, interested of activity, includes sensory deficits, and there are other combinations to redefine more precisely this spectrum. Always is needed a mental health professional at the moment, and of course it's based in the criteria. However, it's not a checklist. Require the observation for the other family members, and of course the diagnostic tools that are part of the comprehensive evaluation and never can be used alone. It's very important to consider the comorbidities because are very common, that include anxiety, specific phobia, conduct disorder, or other depression that sometimes is confused at the beginning, especially if the individual is so young, like 2 years of age. And what is for other nations, include Mexico, the missing diagnosis of this spectrum disorder, or one of these is specifically the lack of awareness of knowledge of the healthcare professional. That's one of the main goals of this lecture, that provide this kind of information, and the cultural factor and the family, and how I say the gender difference that can be more biased to the diagnosis to men compared with women, and the heterogeneity of symptom presentation is diverse as well, masking, especially in females with high functional individuals, that if it's going well in the school, sometimes it's difficult to detect that the individual have this condition, or cultural difference in our regions, in social norms and communication styles, or the access to the health professionals or the lack of training for other specialists, or still general practitioners, or the long waiting list, because this is all a problem. We don't have so much centers to evaluate all the individual with these symptoms, or is detected too late. And now there are trends to get more visible the woman in the neurodiverse world, because we know that there are a delay of the diagnosis, and it's chaotic because it does impact the quality of life. Is now considered more balanced to try to evaluate both in the same way. And there are different advanced diagnostics more than just the tests or the clinical evaluation. Unfortunately, we don't have biomarkers in the worldwide community. They are a genetics testing, but not specific about the condition. And it's increasing the use of telemedicine, especially after the pandemic, or the use of digital technologies and machine learning to include more individuals detecting, basically because it's a lack of a health professional or appointments, and it's one of the strategies that could be implemented in the regions, especially when it's the initial evaluation or part of the comprehensive evaluation to try to detect these trails. And recently the FDA approved this eye tracking device that can help to the diagnosis of the autism using these paradigms that typically have diverse views, and this can be scoring and give the idea about the high risk of this diagnosis. Also, the social network with the surveys or the artificial intelligence algorithm that can be tried to analyze how is the pattern of the person choosing the options. It's possible also have a high confidence of the diagnosis. And this is one of the autism assessment pathway in UK that you see different steps to get the final assessment of patients with the diagnosis of autism. There are a lot of barriers for our countries because starting with the identification, sometimes the patients are adults and it's the first time that they receive the diagnosis, the referral, the screening, the triage, and the assessment, and then the post-assessment support, and take time. And even for developed countries, this is a major problem because the amount of the population that require this service is so huge. You remember, at least 10% of the population is neurodiverse. And there are some principles of intervention. One of the main messages is not only one intervention is unique, is more about the personalized medicine, and that includes many aspects. That's the speech, social skill intervention, cognitive behavior therapy, depends on the level of the condition, educational intervention, occupational, sensory therapy, and of course, sometimes is needed the pharmacological management, and even for the regions, the access to this medication or the supply is other matters because we have limitation for these psychotropic drugs. There are different medical models under the neurodiversity form intervention that can be used or implemented very easily because it's more a holistic approach about this kind of the multimodal strategies. Of course, you need the people to be in training or at least aware about the situation, and that includes the behavior strategy, patient role, school integration, mental health focus, or the limitation that sometimes is possible to see especially in the situation that we are dealing. When you compare the drugs, that is the main core from this lecture, is there are a lot. There are a lot of needs that we can provide for the medical pharmacies, but it's needed. Sometimes one or two, sometimes more, depends of the quality of life that the patient is experiencing. For the local regions, the cultural and structural barriers include the stigma and the misunderstanding about the behavior are considered laziness or with misconduct. Some, in fact, go to the jail. So the delay of diagnosis is very common, not only in Latin America or Mexico, in everywhere. And the research and research gap comparing with Europe and North America, of course, is evident not only for autism and other conditions, and the persistence of the stigma that limit the recovery and the access to the social benefits and the risk of harmful misinformation, because even the social medical community can provide information. There are a lot of fake news in the social media, and we know for our society the false cures that affect the family and the expectation of the individuals. So this paradigm is relevant in Latin America because we try to reduce the stigma, increase the early recognition and intervention, and promote that all the culture are sensitive to these models, and the balance between the myth and the evidence-based communication to get more inclusion for each individual. In Mexico, of course, we have some strategies that can be implemented, and I think some of the region also have very strong initiative to promote the neurodiversity in Mexico. I only choose some of these for the politicians start programs, formal programs for high level education, like in universities, with STEAM programs from the arts, dancing, special programs, sensory lectures. Of course, the awareness of the society together during this awareness day, and the education in the universities to sensitize the society about the neurodivergence. And of course, it's relevant to see that at least for Mexico, if they classify some requirements from the government, they can receive money to maintain the basic needs of these families. In some way, neurodiversity is in a spectrum of the human variation and no disorder. In our region, is key to reduce the stigma, delays, and the resource gap. And the advocacy and awareness are growing, but it need more effort, more time. And the goal is to be inclusive, culturally grounded support system for mental health. That is the new pandemic for this generation. Thank you so much for the opportunity, and it was a pleasure. Hello everyone, my name is Dave.
Well, thank you so much. I believe he's not with us in Zoom, so we have no time for questions because we can't. Right now, well, in conclusion, this is not a luxury. brain science in Latin America is actually a necessity. And I hope all of us got the message that our differences is actually what makes our strengths. And there are a lot of things that are in common between our countries. We have a time for break right now. We're going to come back at 12 Mexico time. I've been asked to inform you that our friends from Psychopharma they want us to try their new app, Aurora, which targets mental health, anxiety, and depression, and you can see that outside. People in Zoom will have their link with their constant participation. Thank you so much. See you at 12.
Well, good afternoon, everybody, both for the persons that are here in the auditorium and for the persons that are connected by Zoom. Let's start the first symposium of this summit. It's called Successful Initiative in Latin America. It's for me an honor to be the coordinator of this symposium, in which we will have 3 presentations. Now let me introduce you the first presentation with the title Latin American Summer School on Epilepsy of the Professor Dr. Esper Caballero from the Department of Neurology and Neurosurgery in the Escola Paulista de Medicina from the Federal University of Sao Paulo in Brazil. Is the professor available? Yes, I see it. Then, Professor, you have the floor.
I'm not listening. Are you listening to me?
Sí, se ven. Yes, yes, we hear you.
Okay, you can see it.
Yes, also.
Okay, great.
Okay.
Okay, thank you very much. Okay, good morning or good afternoon, everyone. I'm very happy to be here with you. And I would like to start by thanking Luisa Rocha, not only to inviting me, but also for her continuous dedication to maintain the flame of Latin American neuroscience alive, despite of all the problems that we have in this region of the world. For me, it's a great pleasure. This is the 3rd meeting I'm following, but this is the first summit. And it's my job now to tell you about the Escola Latinoamericana de Verano de Epilepsia. I mean, is LACE. LACE was created in 2006, and it was a good opportunity because the International League Against Epilepsy created in 2005.
So, Professor, can you put the PowerPoint in presentation form?
I do not need a PowerPoint. This is okay for me.
Okay.
I'll try to move, okay? Because it's just something that you just listen, I'm reading, and that's no special talk or in neuroscience matter. Okay, let's go ahead. So in 2005, as I was saying, the president of the International League Against Epilepsy was Professor Peter Wolf, now living in Denmark. And he was the first to create a commission of education for RELIDE. There was not a commission of education, and we thought that it was a very good opportunity to take this education term to bring to Latin America. There was already a school on epilepsy in Italy, but it was not at that moment under the umbrella of the Education Commission. Our school was the first one to be, and I talked to Peter Wolf, and he said, go ahead, and I start organizing. And the first school was held in 2007. No, 2007 is so far that I'm trying to say 17. But when we prepared this first edition of LACE, it was interesting because we said, we do this, and then we stop because there is no meaning to go ahead. This here is one place in Sao Paulo, is the parliament. Who have been in Sao Paulo or have been to LACE, probably you saw it, is the Parliament of Latin American Parliament located in Sao Paulo. And this is the hand. This hand is the architectural plan of a cover of a book of an Uruguayan the name of the book, most of you, or the oldest people here, would remember. It was the bloody veins of the Latin America, and this was reported here by Niemeyer, the Brazilian architect, and we used this with the all flags of Latin America here to establish the first school, and it remained for the rest of our schools. The objective at that moment was to have an intensive course of 9-10 days looking at translational epilepsy. I mean, going from basic science to the apports that basic science can lead to clinical science, and then back to see how we can say to basic science to answer our questions in clinics. And we aim to receive clinicians and neuroscientists from all over Latin America. The age should be less than 40 years old. All the three languages were used, Portuguese, Spanish, and English. Depending on the lecture, we allow the lecturer speak the language he would like to say. We have that moment, the financing of FAPESP, is the Research Foundation of São Paulo, the ELI, the education group of the International League Against Epilepsy, and now the money is coming via the Academia Latino-Americana de Epilepsia, who was created at first LACE in 2007. So the first was a direct money to ELI, and from then on it was via ALADI, and the Brazilian League Against Epilepsy, and other sources. These are the main sources that we have for support the LACE. We have classes, we have lectures, workshops, practical sections, and the most important, that the participants should be there all time. They have no time for tourists. It's an exclusive dedication to LACE. What we have here is the first edition. It was 4th to 14th February 2007, as I already said. But it was followed because LACE 1 was so good. We gave so much pleasure. The participants were so happy at the end. The teachers were happy that we tried to do the second. And then we did 18 years, except the year of the COVID that we had online meeting. It was a small one. But it was very good because we developed different topics in epilepsy, and going from physiopathology, clinics, and the challenges that we have actually in the contemporaneously. And LACE in 18 years, see, it was consolidated as a very good regional initiative in relation to epileptology. Here are other marks of LACE. This also in São Paulo is what we call the Station da Luz. Luz is the name of the station that in Portuguese means light. It's the same in Spanish, I believe. And these two pictures remind me at that moment that we should bring light to epilepsy. Is epilepsy out the shadows? That was the World Health Organization tribute and campaign to epilepsy at that moment, and still today. And it was a beautiful picture. I said it was also very good. How we organize, which are the institutions linked to LACE. First, as I told you, is the education of ELI Education. My medical school, Escola Paulista de Medicina, la Academia Latino-Americana de Epilepsia, e la Liga Brasileira de Epilepsia are those who decided to support each of the editions of LACE. We also collaborate with national leagues of Latin America. We have two very good examples, the Liga from Argentina, the Liga from Colombia, and some other countries who are sending these students, and they are paying completely or for part of the money we need to maintain a student for 10 days here. The professorship, we have also regional, international professorship, but the care that we had from the first LACE is that we do not want the teachers to come and talk about their personal results. This is a school, and we want them to teach the students, to call the attention of the students for what they are doing. Not a conference, it's just a school, a really talk, okay? The participants are selected by a competitive phase. They have to register, and then we select them. We try every time, every edition, to have a proportion of each country, and also more or less half of men, half of women, and then so on. And we have till today a network of ex-participants from whole Latin America. As I already said, we have lectures about physiopathology, diagnostic, and therapy of a topic that we choose. We have also workshops about semiology, case discussion, activities of problem solutions or resolutions, if you want. The emphasis is always translational, but with a regional perspective. What is important to know about epilepsy in Latin America? Which are the differences among our countries and the rest of the world? This is quite important. And the available treatment, the available diagnostics that we have in different regions in Latin America. And the students are divided in small groups of 6 or 7 students, and they have during LACE, in the evening, normally during the evening or even the whole night, they have to elaborate one research project based on the topic of each year, and they have to present on the last day concerning all phases of the research project. From the title, they have to think about the question, which would be a good question for this year, and which is literature. They have to prepare methodology and objectives, and also, when possible, the money that we need. And they do it. Everyone did it fantastically. We have very good projects there. And this is the place where Santa Monica Hotel, Villa Santa Monica, where we have LACE since the first to the last edition this year. The topics that we selected are just some here. LACE 1 was epilepsy translating basic knowledge into clinical applications. LACE 3, epileptogenesis in developing brain, basis for treatment and prevention. LACE 4, epilepsy and time. The polio, and the last one was modifying the natural history of the epilepsies. I should say that I've been learning a lot during each LACE. The impact and results of LACE. We have already more than 1,000 Latin American participants at LACE, what for us is a huge number of people that have been visiting us this 18th year. I think that we create a regional network of epileptology. The people, now they know each other. Even those from Uruguay or the south of Chile know people from Mexico and from other areas. This is very, very useful. We have integrated basic science and clinic. We develop collaborative projects, and we we also receive some engineers working in EEG, or some nurses, or some psychologists, or even teachers from very basic education to learn, because they want to know what is epilepsy is, okay? And we also take the opportunity to diffuse the good practices, diagnostic and therapeutics. And we would like to improve the quality of life of the people with epilepsy in our region. At one moment, I decided to stop to have LACE this year, and I discussed with all the organizers, the people who have been helping me for more than 18 editions, because I think that LACE needed to change. It needed to change. I'm quite old now to do all changes. I think that after 18 years, we need to have other objectives, other kind of teaching, other kind of meeting. And we have not selected, not this. Patricia Braga from Uruguay was now, she became the responsible for the next LACE. I hope that she remains for the future, not only for the next LACE. And they organized the next edition, a new edition, a different approach from Guadalajara, Mexico. And we are waiting to see what this young generation could do much better than. I'm sure that they are better than us now. They are the situation of the technologies, everything is much better now. To contact LACE, you have the site, is lace.education. There is also some space in the ELI congresses and schools. There is the site ELI Education, and there are some social network with ELI, mainly Instagram or Facebook. I don't know where the students are preparing. They are doing these things, but I cannot follow. So this is a very short education. I'm concluding now, and because LACE for me and for most of those that were able to participate, we all were very happy. Each year receive 60 participants from Latin America and 60 different people. They were so engaged all time. They were present, they were tired, they were very, very tired, but very enthusiastic. And the last day, I mean, talking to me? Oh, no.
No.
No, okay, I was listening to some songs here. Okay, and just to say that the last day when they present the research project, it was so fantastic because we could see how they grow during these 10 days. They were talking like a teacher, giving us very good ideas of research project. Of course, they belong to us, not to us. To them, not to us. I mean, the project are them. They can do, they can read. And we were also very happy. You know that there is one European journal of epilepsy. The name is Seizure. And 2021, they invited us to have a selection of papers from students and or teachers to have a special number of LACE. It was done, and now they are, and the last 2 years, we decided to have some stimulated by two people, one teacher from Lithuania and another from Colombia. They decide to have pictures of the students' designs, you know. They bring color pencils and papers and say, what do you think about the topic of this LACE? And the students are designing. And the Seizure decide to take this design to put in the cover of each number of slides. What for us is we are proud of doing this, you know. the students are very happy to have one of their projects in the front page of the journal. And I think that it's always very good when you have a meeting in several countries of Latin America to meet the past students. They are fantastic. They are doing quite well. Most of the past participants are now engaged in the national leagues. Some became president of the national leagues. Others are contributing in several. I think that is a change, completely change in the Latin America perspective of epilepsy and the care of the people with epilepsy, who are the main objective of the school, is to improve their lives and the understanding that they have about the pathology and how to be treated and where and who they have to select to treat them. This is very useful in every part of the world. So thank you. I think that I have some time for questions, but you can write to me or write to Lasse if you are interested in going in or sending your students and so on. We are very happy. Thank you.
Yeah, thank you for you, Professor Caballero. It's time for comments, questions from the auditorium. Any question, comments? Yes.
Thank you, Dr. Esper. I would like to make a comment and a question. A comment, I was a student in 2024. I don't know if you are hearing me, Dr. Esper. We are not hearing you. Oh,
Okay, and now I'm talking. I do not know who is talking. I would like to know your name.
Oh, I'm Daniel, Dr.
Oh, Daniel, how are you?
Yeah.
Okay.
I would like to add a comment and as an experience that Lasse is actually a life-changing experience. I would like to add to all that you just said that we have the privilege to interact constantly with professors, with students in a very close environment in a favela in Brazil, which is a very interesting experience. It's also a highly demanding cognitive task because we were always discussing the projects, if not during the lectures or the workshops. So I think it's a great experience that everyone should, interested in epilepsy, should have. And my question is, what does it take to start these kind of initiatives? Maybe not in epilepsy, but in other areas of neuroscience, as we are here together to discuss possibilities to make neuroscience in Latin America to move forward. What do you think it takes to improve and to develop some other schools like this?
Yeah, this is not an easy task. As I told in the beginning, that it was a window of opportunity that we had. It was the presence of Peter Wolf as a president of the International League. I think that the role of the president of the associations is extremely important. They have not to think about their position as president. They are beautiful, they are wonderful, they are potent, but they have to put things outside of them, you know, open the arms and look at the future. This is very difficult in scientific societies because we are always so proud to be there as president that we forget that you have to serve and not be the president, but to serve the community. So I think that we have to find this president in other neuroscientific societies. I think it's very important. We have here in this room today very important people linked to different associations, and go there and promote education in Latin America. This is the most important thing that we have to do. Promote scientific education in Latin America. We have to do that. Luisa is doing that. I was thinking to say that from Laura, the first lecture this morning till now, Daniel, the last one that I saw, all taught us a lot of different things. It was so impressive for me to listen. And I was trying to propose to Luisa to do a meeting like this every 6 months, because it's so good opportunity to understand other areas that we are not focused on. I think that this is so important for everyone working in the brain, to listen from the problems of the children and the work she did with 2006 children. This is fantastic to know. And Daniel talking about the neurodiversity, it's so, so broad. Neurology is so broad, the brain is so important, as we know, is doing so many things that we do not know yet everything, and that we should do that, educate Latin America. This is the best thing that you can do in science, and for losing this feeling that we always have to be part of a second-class society or world. You have to think, don't have this feeling, just do our best for improve the quality of our countries or part of the world.
Thank you, Dr. Esper.
Thank you. Any other comment, questions? Okay. Then we will pass to the next presentation. Now I will to introduce Professor Agustin Ibáñez, who will to present us the talk titled Latin American and Caribbean Consortium on Dementia. Professor Ibáñez is the coordinator of the Red LAT multi-partner consortium to expand dementia research in Latin America from Chile. Professor, are you ready? Okay, go ahead.
Can you hear me now?
Yes, yes.
Okay, thank you so much for the invitation. It's a great pleasure to tell us a little bit about our two consortiums. It's a great pleasure to learn more from colleagues working in Latin America or for Latin America. I will share my screen. Can you see my screen right now?
Yes, yes.
Perfect. So those are my disclosures. And today I will try to summarize what we are doing in these two consortiums, why Latin American boys are really important in the, I would say, in global studies of brain health, but especially in the world majority. Then I will introduce the Red LAT consortium, that is the research-oriented consortium that we have developed together with 13 centers across 7 countries. We will talk about multimodal diversity in terms of genetics, phenotypical presentation, and environmental drivers. And also we'll focus on whole body health and exposome. And then I will present some initiatives that we have developed beyond science for the region. So the first question is why we need brain health research in Latin America, right? The quick answers comes from South Africa. This is Johnny Miller, a big friend of mine, that he was a common photographer until he became very famous with this picture. This is Johannesburg, one of the largest inequal cities in the world. So if you're born on the left or the right side of the picture, your socioeconomic life is going to be completely different. But it's not only socioeconomic, it's not only your pocket, it's your health. Let's say Juana, same person with the same initial conditions, same genetics, living in the, this left or right side will evolve through a much more stressed brain, the allostatic interoceptive network, if they live in the right side. We'll have more cardiovascular factors, cardiometabolic problems, cardiac problems, and dysregulation between body signals and the brain. We'll have, more accentuated biomarkers in terms of cardiometabolic, immune, inflammatory, and even neurodegenerative parameters. And also we have less, environmental support to learning to self-regulate and to, developing brain skills. So This is the why, and I want to present this example we published recently, the Colombia case. That is a case that I think that shares a lot of challenges in our region. So Colombia, present a really complex scenario combining narco trafficking, internal displacement by violence, but also, a lot of uncontrolled violence from Venezuela, a lot of socioeconomic inequality, poverty, and all these things come together. And the impact, the toll on the brain health and the mental health is totally ignored. So this case that is perhaps a little bit extreme case in Latin America, is happened across the region. We have been working trying to show that Latin America is a very diverse region in both sense. In traditional sense of genetics, we are a mixture from native African, European populations, sometimes Asian also from Brazil, for example, but also huge heterogeneity in terms of access to health, education, socioeconomic inequalities, and many factors that have been making a case. So we already shown that the typical risk factors for healthy aging are completely different in Latin America, and structural inequalities have a stronger role, more than classical factors like age or gender, for example. And when we try to fit the models that come from Europe and US into Latin American data, you will see that there is I will use my pointer here, there is really the models work in a more weak version, because models are trained with different data. That's the simple history. Then we have shown that we need to do a lot of computational things and addressing socioeconomic factors. I will come back about that. We also have shown that the typical phenotypical association between brain and complex behavior like social cognition in healthy aging and dementia are behaving in a different way when you consider underrepresented populations. And the most important thing, how environmental factors interact with genetics in a region, is completely unknown. There are so many insights from Latin America for genetics, but how those factors interact with environmental exposomic factors is absolutely unknown. Given that, we created the Latin American and Caribbean Consortium on Dementia and the Latin American Brain Health Institute that is based in Chile, but coordinates 13 centers across 7 countries: Argentina, Chile, Colombia, Peru, Brazil, and Mexico. And we have been working to empowering the voices of different emergent leaders in the region. So we created the Red Lat Consortium, which is mainly NIH-based consortium, but also supported by many other stakeholders that try to address 2 things: the heterogeneous, genetics that we have, but also the heterogeneous environmental factors in terms of social determinants of health, socioeconomic factors, and as I will show later, also exposomic and environmental factors in a more expanded sense. We have been published more than 250 papers nowadays. We have developed multiple educational programs, including training for diagnosis of dementia for clinicians, psychiatrists, neurologists, training for neurocognitive assessment. We are trying to expand our efforts. So one thing that we have discovered that the heterogeneity, when we talk about Latinos, in US, even in US, sometimes we mix Latinos and Hispanics that are completely different population, not only in terms of admixture in genetics, but also in terms of lifespan experiences. We have shown that the heterogeneity within Latin America is huge, and you need to develop, strong computational models to deal with that heterogeneity when you look at neurocognitive profiles. Also, we have been pushing the idea that, we need to bring more information in the biological embedding of the socioeconomic factor. Most of the online data sets in the globe are using, just education or even less years of education, but what about profession, income, neighborhood, access to health? This is hard to get in global data sets, and it's a we already know that impact multiple epigenetic, cellular, microbiome, peripheral, cerebral, and cognitive factors. So we need to change a little bit this history. We have shown, for instance, that both qualitative and quantitative education have a huge impact in the brain, both in terms of structure and function in aging, but also in Alzheimer's disease and frontotemporal dementia. We also have mentioned that more complex social exposome, like the combined factors regarding quality of education, the education of your parents, the nutrition, the financial status, very early stage aspect like childhood labor, or traumatic childhood experience, they have a huge impact in the long term, in the aging and in the risk of dementia. And this is happening at neuropsychiatric level, at cognitive level, at functional level, but also at brain level, at the structural and functional level. This is a paper that is going to be published probably next week. And we also have been shown that, very simple metrics like inequality, the country inequality really modulates the structure and function of the brain. So the larger the inequality of a country or a city, the reduces the brain volume and the functional connectivity, once again, in aging, Alzheimer's, and frontotemporal dementia. But also, plasma biomarkers are perhaps one of the most important initiatives nowadays for accelerating diagnosis in a very affordable way in global settings. But, we have shown that for Latin American samples that are, more complex disease with multi-proteinopathies, with larger cardiometabolic, confoundings and poorer whole body health. We have shown that you need to, at the very least, to combine clinical metrics with a standard ATM framework, like trying to assess tau, amyloid, and neurodegeneration. And when you combine this metric with brain and cognitive measures, you really can characterize the patient. Only with plasma markers is rather limited. We also have shown the huge impact of the families in the region. So, for instance, this is a typical presenilin family originating from Francisco Lopera's group, but also, programming mutations. As you can see, you can do a full clinical trial with just a family. So this is another characteristic of our family, especially in the Caribbean region. So large families, we have found many mutations, and still we have more than 200 families with a typical genetic presentations, but we don't know yet what are the mutations. We are starting to do, clinical pathological associations, long reads, and already doing, ancestry analysis showing how the ancestry really influence genetics. Other groups that I have the pleasure to collaborate, like Janitza Montalvo with the Latin American genetic group in the psychiatry domain, have started to do exactly the same, showing that, the huge diversity in terms of ancestry impacts, the presentation of clinical mental health conditions. We also are developing transferable methodologies from MRI and fMRI to EEG, because EEG is very affordable, and sometimes the data quality of our MRIs in Latin America is not the best. So we have using biophysical modeling with fMRI and showing that with EEG or EMG you can have quite similar results, very robust results with a much more affordable metric. We are doing the same here, for instance, comparing nonlinear irreversibility nonlinear metrics, showing that both fMRI and EEG have a comparable performance to characterize patients. In another R1, we started to develop more affordable metrics to characterize the patient, so we use natural speech analysis, and we already have shown that just with 10 minutes of recordings of the voice, we can build more than 900 features for each subject, and we can use this feature connected, with clinical, cognitive, epigenetic, multimodal neuroimaging markers to identify not only diagnosis but also severity, but also subtle impairments across different ADs or frontotemporal dementia presentations. In another one with Kunghu, we are starting to use wearables. So a simple device that can track your movement, can have a broad estimation of sleep quality, and you can also, use it to do it circadian assessments. With these approaches, we have been shown that very simple things like pulse rate is really associated with faster cognitive decline in older adults. I think this is a kind of method that we need to improve to make it more scalable in the region. Also using 4 channels during EEG recordings and using high order interactions, we have been shown that we can predict the conversion to Alzheimer's. In another one, we have tried to investigate more in depth the biological embedding of social inequality. So for that, we use social epigenetics, and we use both, organ clocks as well as methylation and second and third generation of aging clocks. And we are now starting to show that, like second generation clocks like PhenoAge or GrimAge are really good to predict AD conversion, for example. And now we are in the process of developing the renewal of our consortium, where we want to focus in larger aspect of the exposome, introducing more levels of social and physical factors, how they impact in the brain, and also better computational models using brain clocks to better develop this approach. So the first step we did for that is showing that using convolutional neural network with both EEG and or fMRI, we estimated the brain age of the participants and then the gap between the brain age estimation versus the chronological age. So you can identify individuals that are younger or older in the brain in comparison with the chronological age. Using this approach, we have been already shown that, if you are female, you have accelerated aging. If you come from the South, from Latin America, you have more accelerated aging. If you have dementia, you have more accelerated aging. But if you are a female coming from Latin America and having dementia, you have the largest accelerated aging. In this study, we also started to explore some socioeconomic and environmental exposomes showing that there was an effect. But in this paper, across 40 countries, including Latin America, we were able to develop a new metric of biobehavioral clocks in which we estimate based in the cardiometabolic, biological metrics, and cognitive metrics, the expected age for a person. So what we found is that Africa have the largest accelerated aging in Egypt and South Africa, followed by Latin America, then Asia, and then Europe. First, Mediterranean Europe and then Northern Europe. So Northern Europe have the healthy aging, of course. But we combined different, protective factors, risk factors, but we also included multiple exposome factors. That include pollution, for example, as a physical exposome. That include also, socioeconomic inequality or migration as social exposome, but also we include a metric of sociopolitical instability, and we've shown that even the stress triggered by sociopolitical instability accelerates the brain. So I think that this is kind of model that we need to develop, tailored model that capture, the embedding conditions of the brain health. In this follow-up study, we are doing the same, but looking instead of biobehavioral clocks, we are now looking at brain clocks, but now we are combining 72 different exposomes from air pollution, green space, quality of the soil, of the water, temperature, precipitation, climate disaster, as well, typical socioeconomic metrics, structural inequality, gender inequality, unemployment rate, GDP, but also this metric of democracy, right? So we have been shown using nonlinear methods that, when you have multiple exposomes, adverse exposome combined, you have a huge exponential increase of the brain aging in comparison with the impact of the individual exposome. That means that the regions that are, underserved in terms of multiple socioeconomic, social, and physical exposome represent a more challenging environment that will accelerate the brain aging. For that, we are proposing computational models that can, model multi-levels of the brain's cytoarchitectonics or neurotransmitters integrated with functional dynamics, but also incorporating the whole body and the environmental external factors in order to develop better computational models that can address this multimodal diversity. This is something important not only for disease, but also for resilience. In this recent paper, we redefined resilience as a truly biological, but also cognitive, psychological, and social phenomenon. As you know, there are mutations like the Colbos mutation or the Chris-Charles mutation that protects you against dementia. But the environmental factors can also protect your brain health, as well community resilience. So, if we understand the resilience as a way to manage the lifetime cumulative impact of stress, you will see that the way that your social networks, your family, your community distribute this stress and help to cope with this situation in the long run can have a huge biological embedding. And this is why I think also we need to understand how these synergetic factors that happen, from exposome and social inequalities through whole body, have a really more synergetic dimensional and specific analysis. So they are not just important for brain health or for dementia, they are important also for stroke, for epilepsy, but also for mental health like depression, anxiety, and much more. This is, again, why we need to develop tailored, locally informed models. We need to stop to think in the mind and the brain as a universal thing, something that I call it, a golden cage. Then when you control all the external factors and just look at the brain, we need to understand the brain and the mind in the wild, in the environmental conditions. So this is that we are proposing basically to try to understand how the environment creates an imprinting in the whole body health, and at the same time the whole body health have a huge impact in the brain model. So for that we really need different computational models. We need to understand the brain within the body and the body within the context. So this is also important from some global initiatives like the One Health, now led by the WHO approach, in which you need to understand how the health environment not only impacts the environment, but also the body and the brain, and how we can try to integrate physical and social exposome together with the internal exposome in terms of biological responses to those environmental demands, including epigenetics, allostatic overload, or inflammation. And the allostatic interoceptive process are a relatively good model to try to understand how the environment create a continuous pressure for the organs to respond, not only at the brain level, but the organs levels, creating a huge adaptation. But when the environmental demands are sustained over time and are too much strong, the allostatic factors start to be more cumulative and associated with multiple physiopathological pathways. This is also why when you look at neurodegeneration patterns across the global south, you will find different variations in terms of not only genetics, but neuroimaging, plasma, and other biomarkers. So this is what we are doing in research, but we also are trying to push scientific diplomacy and regional networking in order to, creating a local voice or a regional voice that not only, contributes to the global agenda, but also create their own agenda. There are many questions that are usually neglected because they are not in the global priorities, but are really important for Latin America. So creating multiple voices, I think that is important, empowering the the voices of the clinician and the researcher is also important. We have started to do something similar in Africa with the Yaoundé Declaration. And I think that we still need to collaborate much more between Latin Americans. I would love to explore more collaboration with the fantastic work that you are doing. We also have been working with the Inter-American Developmental Bank to improve and trying to develop approaches for diagnosis of dementia that preserve both the local tools, but also that can be partially harmonized in more global settings. And also we have been training many people, so we created the two first brain health courses together with the Inter-American Developmental Bank and the Global Brain Health Institute. We created, toolkits for supporting health diplomacy, to empowering scientists, to learning to speak to policymakers, but also to connect with different fields. We also want to work with our communities, and this is a wonderful example of musicians that are street musicians with terrible histories, living in the streets, being homeless, but wanted to change the life of young people. So we work with them, and we were able to make these children playing for the first time in a scenario, while we teach them about brain health protection, while we teach them about prosociality, and we have very nice results, with a small thing. This is why I think I am preparing this paper in the new Lancet series for arts and health. I think that we can work with the communities to reduce stress, to improve self-organization, to create better environments for learning, and for learning about the future of your health, of your brain health. That is something that can be definitely, teach. And I think that there is a lot of opportunities there to work with the communities to create more prevention. So the Global Brain Health Institute, GBHI, have been very instrumental in helping us to coordinate. Our consortium have more than 26 fellows across different Latin American countries working in the Red Lab Consortium. And that's it. I hope that you have a grasp of what we are doing, and I will love to explore any collaborations with you. Thank you.
Thank you very much, Professor Ibáñez. Some questions, comments from the auditorium? Okay. Yes.
Hello, Dr. Ibáñez. I have a question. Do you think if we take in account more the brain functioning in the wild, it will be possible to obtain more support from the politics? Do you think? More support from
policy?
From the governments.
Oh, yes. It's a great question. Sorry, I don't know your name. Thank you for your question. I think that this is essential, right? If we are able to bring the brain, as a way to show that inequalities, for instance, impact health in a very clear way, in a very, critical way, I think that the politicians at the very least will pay a little bit more attention. Also, if you consider, let's give the example of dementia. Dementia, if you combine indirect and direct cost, it's one of the most costly disease, more than cancer. So I think that first of all, we need to bring neuroscience out of the laboratory. Not only, of course, the basic neuroscience is essential. I'm not trying to say something against that. What I'm trying to say is that we need to connect these findings with, other aspects, and population neuroscience, and in particular the exposome, like, working with people that work with climate change or environmental factors or pollution or socioeconomic inequalities, by showing the biological embedding of these factors, in terms of accelerated aging, epigenetics, and many other markers. I think that we can make more evident that if we don't change the structural inequalities, we won't have healthy brains. And the healthy brains are the most important capital of a nation. It's the most important added value that a nation can have. So I definitely think that is a way to try to push the governments. Of course, it's not an easy task, and it's not a short-term task.
other comment? Okay, yes.
Thank you, Dr. Agustin. Daniel, and from Colombia, and I really like the example that you gave at the beginning. And just in context, Colombia has the second internal biggest diaspora after Syria, and a history of violence and, well, many other things. My question is, do you think these past traumas, this big diaspora in the 90s, can be inherited to the further generations? And if so, how can we deal with these consequences of early traumas that passed many years
Sorry,
ago?
I couldn't hear well your question. Apologize. Anybody can repeat the question because I was.
Sure. Yeah, in other words, I would like to know if you consider that prior adverse situations like the internal diaspora of Colombia can be inherited to the consequences, epigenetic or, well, biological consequences can be inherited to the further generations, and if so, how can we deal with those consequences?
Yes, sorry, I don't know. The sound is very poor here. I am in a hotel with bad connection. I guess that you are asking if these terrible environmental conditions like violence or other can be impacting and in a biological way the next generation or something like that.
Yes, exactly. Yeah, and if so, how can we deal with those consequences?
Yes. So yes, definitely. As you know, epigenetics have been shown that the effects of environmental stress in animals can have 2 or 3 generations levels. There are some poor studies, I would say, of intergenerational transmission of violence. I think that we need more evidence, but in principle, with all that we know about epigenetics, we really it's happened with Colombia. In Ireland, where I work, second and third generation of ex-combatants tend to have high level of drug consumption or alcoholism. So I think that this biological embedding of the environmental experience that is transmitted through generations. So what we can do, we need to do much more better intervention in that case. I think that Colombia is an example. The government is really worried about has been worried for many years since the peace agreement, trying to develop programs for integration. But these programs tend to fail. There are an important percentage of the people that fail because the interventions are short term. You cannot change the impact of learning violence for 2-3 generations with 6 months intervention. So what we need to do is, again, to develop long-term interventions, to develop multi-component interventions. It's not enough, working with trauma. You need to work with the whole mental health spectrum, but also you need to address other things like, social conditions, social life, exercise, quality of life. And this is a very big challenge. I think that it will take a long run until we understand that you need to do long-term intervention to try to face these adversities.
Thank you.
Okay, thank you. We have 2 requests of question or comment from the Zoom's participants. First, Professor Maria Castelló. No?
Sorry, I cannot hear nothing.
Angelica,
if somebody can write down the question, it will be more easy for me to answer.
No? Okay.
Or maybe...
Yes, we have a request of question or comment from Angelica Osorio in the Zoom participants. Angelica?
You are muted, Angelica.
Angelica, we can hear you. Please.
Muted. You are muted. Your microphone is
maybe in the
off.
in the control room. Can you activate the microphone? Okay, yeah. Okay.
Yeah, that's okay.
Yes, I can hear.
Thank you. My name is Angelica Osorio, and I work in a university in Puebla City. And more than question is an invitation, and I would like that you give us a kind of this presentation to my students. They are they are studying medicine. So I would like to have your email, maybe.
Sure, of course. I will write down my email, and I will be happy not only to give a talk, but also, to provide with initiatives. There are many, not many, but there are some important initiatives that may be of benefit, and also look at the GBHI, the Global Brain Health Institute, that provide fellowship for people that want to fight, brain inequality. So I think that and GBHI can receive, musicians, scientists, policymakers, all type of profiles that have as goal to reduce inequalities in brain health. I am writing down my email.
Thank you so much.
Okay, thank you. Then let me now introduce the last presentation of this symposium. Thank you. Yeah. Is for Professor Daniel Olazábal from the La República University in Uruguay. Daniel is also the representative of LATBrain in Uruguay. And the title of his conference is Challenge of Interdisciplinary in Brain Science, Building Bridge. Okay, Professor.
Thank you. Thank you, Roberto. Can you hear me? Yes? Okay. I am, thank you for the invitation and giving me the opportunity to share some comments and some thoughts about how to do interdisciplinary research. I hope this talk in some way can help you, some of you that might be interested in doing this kind of research. Next. Okay. Well, why collaboration across fields is both hard and essential for understanding brain function? Well, we all know that the brain is one of the most complex systems known. No single discipline can encompass its full understanding. Neuroscience is nourished in general by many other disciplines, and without the integration of these disciplines, we miss important parts. So what is interdisciplinarity? Sometimes it's not easy to distinguish from multidisciplinarity or transdisciplinarity. This is a more recent approach. In multidisciplinarity, there is a coexistence of disciplines. Different disciplines work in parallel to address a problem, but they don't really integrate. They just approach the problem from different point of view. Interdisciplinary means integration, so the discipline integrate effort from the start of the research to the end, as represented in this diagram of the right side. So how to go from coexistence to integration? Well, in general, collaboration should start from the beginning of the study design, and continue until you get the results or eventually your application, if it's that what you are looking for. So I want to give you, for example, some examples that are not interdisciplinary research. For example, someone that say, oh, I included you in my grant proposal. So I maybe the person from other discipline doesn't even know exactly what he or she is going to do in your grant proposal, or maybe even not be interested. Okay, maybe might not be interested. And so that is not multidisciplinary or interdisciplinary research, of course. The second example might be multidisciplinary research, but it's not interdisciplinary research. I study A and you study B in a certain study. There is no integration. It's just a division of work, and you keep using your discipline exactly as you always do, and the other the same. So how to go from coexistence to integration? Well, might be many ways. I just mentioning some things I think are good, like for example, creating seminars and academic exchanges that lead to interdisciplinary experimental design and research. So you have to create these instances where you share your research and the research of other people that in some way are connected what you are doing because you have common interest. But in this way, you might start to learn about the other discipline and the way they address the problems and what are the interests, what are the differences. And once you get familiar with the way they work and what they do, that is going to be more easier to start to think about common research. So interdisciplinarity sounds great, but of course it's not easy. Disciplines has many times different language, most of the time, and terminologies and jargons. For example, what is model for one discipline, like a computational model, for other might be mathematical model, for other an animal model, for other a model to predicting a behavior. You might be talking about different things when you use a word. And also, there are some words that are almost very popular in one discipline and are almost forbidden in other disciplines. You know, you see a small, innocent word, but when it's heard by another researcher from another discipline, it's like waking up a demon. People really hate some words. So you have to know that, you have to be aware that that is going to happen to you, and something that you are going to say might happen to your colleague from other discipline. Sometimes there are different goals. You know, some people might be more interested in getting application rather quickly, and others might be more interested in understanding the mechanism. They both are fine, but once you want to start a collaboration, you also have to be open and patient, because you must know that the goals might be different. So another problem that maybe some institutions or some countries have already resolved is that we don't have, I didn't have a multidisciplinary or interdisciplinary or cross-disciplinary training. I mean, I have tried to learn in my career from other disciplines and try to train myself, but still there are many institutions that do not have this kind of training. So this is another difficulty, because we are not familiar to do this and to learn in an interdisciplinary way. Another problem that maybe some institutions have also or already adapted to the new times, but I know many institutions that do not, and they are very rigid. So you might be doing interdisciplinary research and publishing things that are interdisciplinary or multidisciplinary, and then you apply it for a job in one department, suppose Department of Physiology, Department of Biochemistry, and maybe your study has a little bit of physiology, but it's not clearly a physiological paper, or a little bit of biochemistry, but it's not clearly a biochemistry paper. So some departments do not consider your publication. That happened. So in that way, you cannot encourage interdisciplinarity, and you might be in the situation where I fit, because I study this, but doesn't fit in clearly 100% in physiology, doesn't fit clearly 100% in biochemistry, doesn't fit in, I use different disciplines to try to understand a problem. Well, that happened also with grant proposal evaluation. You might present an interdisciplinary grant proposal, and the reviewer might not be prepared to evaluate the different aspects that you included in that proposal. Some institutions might have also problem with budget sharing because they are not adapted to, for example, share money that were provided to the grant between institutions. That might be a difficult barrier. And also, sometimes we don't have the space and the time to get together with other colleagues to exchange about our research in an interdisciplinary way. So this creates many tensions in collaboration. For example, you can work with psychologists working with geneticists, and the psychologist might want to interpret the behavior, might be focusing in going in that direction, and the genetics want to focus on the genetic predisposition, and one is going to try to make their view more important than the other, and vice versa. So neuroscientists and sociologists is one of the most difficult communications because, some people say, no, epistemologically I cannot collaborate with this. And sometimes you don't even talk to the person. It's just a preconception. They think they know what you think, and most of the time that is wrong. And the same for neuroscientists who say, no, the brain explains everything. You can study social neuroscience and explain everything through that. We don't need to collaborate with. Well, this tension need to be, you must know that are going to happen, and you need to work to understand and hear the other side, because the other side has many things interesting to teach you. Example number 3, neuroscience and artificial intelligence. Maybe you think that this is very good and common partnership, but it can not be. And someone could say artificial intelligence is simulating the brain, and a neuroscientist say, no, artificial intelligence works in a very different way. The brain is much more complex than that. So again, even when there is enormous synergy that can increase the quality and advance science, many times there are tensions and conflicts. And we don't have too many shared forums for dialogue. So, but why we need to integrate? What is the power of integration? In the last decades, mainly with the example of brain initiative in many countries in the world, we have learned a lot about the brain. We have improved treatment, and we can now detect earlier many brain disorders. At the same time, technological innovation solutions have improved the accessibility to many things in people that are limited in their motor capabilities, they're paralyzed for different reasons, and it's probably promising many improvement in many conditions, diseases, neurological diseases, etc., as you probably may know. For example, optogenetic is a clear new finding consequence of interdisciplinarity, where genetic, neuroscience, physics, and engineering work together to create these capabilities. And I want to mention some local and regional examples that where I have been involved. For example, in the last 2 years, we write a proposal of a brain institute in our country. We gathered for this first time, this has not happened in our country before, investigators for engineering, chemistry, robotics, social science, psychology, neurology, psychiatry, and many other disciplines. We established a dialogue for the first time that is very promising. In fact, we wrote a proposal that was very well considered by experts of Latin America when they evaluated that. And sooner or later, I'm sure we are going to have this Brain Institute. And the most important is that to do these things, you need to approach other disciplines and work in an interdisciplinary way. And also create the environment for everybody to be happy of participating, right? This year, maybe 5 or 6 months ago, we also created, with other colleagues, a Latin American Caring Brain Network that also includes psychiatry, psychologists, social scientists, molecular biologists, neuroscientists, and there was an immediate, an immediate good reception of this idea. People love it, and everybody want to participate, and immediately started to create new opportunities of collaborations, invitation for seminars from one to other. And again, we hope this is going to grow and create in the future collaborations, and in an interdisciplinary way. We hope we start to generate interdisciplinary proposals where many different people from Latin America and from different views will participate. And I cannot change next. I got okay? And I have to mention that those examples that I just mentioned were almost 80–90% a consequence of the existence of LATBrain that has had an enormous impact in, I don't think only in Europe, I'm sure in many other countries of Latin America. And one of the goals of LATBrain is also promoting interdisciplinary and transdisciplinary collaborations and to create many networks. There are other networks, a part of this, that have been created. I think it's in epilepsy and new centers. So I need to highlight this because the other two previous examples were consequence of this. Well, practical strategies maybe can help some of you. If you don't have in your institution cross-disciplinary programs on rotation, that might be a good idea. For example, for basic and clinical collaboration. You know, there might be programs where clinicians need to spend some time in a laboratory to learn how you work, neuroscientists, for example, work, or how a molecular biology work. And the opposite too. Molecular biologists or neuroscientists that are working in the lab or in biotheriums might spend some time with clinicians, seeing really interaction between medical doctors and patients, and seeing how they address, and that will help. That will help the student to understand better what is needed to do research. Because if you are in a lab, it's difficult to visualize what is the need of a clinician. And the opposite, if you are a clinician, it's very difficult to see how many opportunities laboratory scientists can provide. Try to create spaces for collaboration and dialogue, like I mentioned, workshops, symposia. And at the institutional level, it might be good if they have not done that already, to create a grant specifically focused to interdisciplinary research, scholarship specifically focused to interdisciplinary programs and students, and try to find ways to recognize the people that are really working in interdisciplinary research. This is a collective act of scientific courage, because you had to overcome all these barriers. Building bridges sometimes also require removing barriers, including pride. I was talking the other day about a very prominent scientist, now retired, in Uruguay that in a meeting that we had with him, he said, but you don't forget that disciplines or institutions also have pride. It's like they really have pride. And when you want to join disciplines to work together or institutions to work together, they are also going to be the impression that the institution want to have a most important part in the components that the proposal are divided or composed, and you might need to think about the common good. You may have to put aside your personal pride, your discipline pride, your institutional pride, and maybe also your national pride. Because in this way, I think we all will benefit ourselves from having better science in our institutions, in our countries, at the regional level, and hopefully at the global level. So I encourage you, those that want to do interdisciplinary research, because someone may not be interested, that's fine. You can keep working in your discipline, we need that. But this is really important to start to consider other discipline, because it has been proven that increase the quality of your research, and it's very important to solve your problem. So I encourage you to keep building bridges, and this was the message that I wanted to share with you.
Daniel, question or comment from the auditorium? Yes?
Well, first of all, thank you so much for this line of investigation. I'm very interested in the line of interdisciplinary research because sometimes as a college student, it's very obvious for us that our educational system lacks of this important area. So my question for you is, what do you think about being interdisciplinary as an individual? I mean, do you think scientists should focus on only one area of investigation, or do you think that could dominate more than one? And if you think so, what would...
I didn't understand the last part you say. Just the last two sentences, maybe.
Do you
Can
think...
you speak a little louder?
Yeah. Do you think that scientists as individuals should focus on just one area of knowledge, or do you think that it's possible to dominate more than one?
I'm not sure I understood. Maybe you can repeat in Spanish, but you are talking about if you can handle like more than one area of knowledge?
Yes.
As a student or as
as a researcher and as a student?
Oh, well, I think that depends on you and what do you think about dominating an area. I mean, once you go beyond your comfort zone, so you move to another discipline, that's why you require courage, because you are going like to start over again. But if you do it early, maybe you can keep working in what you are doing, but you are moving into a newer area. I think you can perfectly well to incorporate the big component of both, or maybe three areas. For example, I don't know, I'm not a genetist, but I have used genetics in my study. I'm not a biochemistry, I have used things that are part of the biochemistry. I'm not a social scientist, but in some studies in human that I published, I had to use many social contexts like demographic and early experience variables. And so you at least, I mean, you cannot be an encyclopedic expert in all the areas, but you need to, you can learn to understand what you need to consider and what you can use from all the other disciplines that can be good for your goals, that whatever that goal is. And in education, I think that training that helps students and recognize the effort of students to train in more than one discipline and integrate them should be encouraged and rewarded. Because I think it probably require also some, like, you need to be used to think in that way, because some other people just don't think in that way and like to, you know, keep in their very enclosed space that they feel safe and they know very well, probably. But the reality suggests very strongly that if you do not open a little bit to new things and new disciplines, your probability to contribute might be less powerful, let's say, I don't know.
I have a question. May I?
Oh, yes, ask the question, please.
Sí, Professor Esper, can you ask? Okay.
I can go ahead?
Yes.
Okay, thank you. Are you listening to me?
Yes.
Okay. I believe that you know quite well the work from at least 20 years ago, talking about converging knowledge and converging technologies for the society. And this was two authors writing about the first idea of grouping complex topics to work with all knowledge available. It was more or less the same that you're talking, but they tried to use the word convergence and not transdisciplinary, just to avoid to have the word discipline again, because all topics that you follow in the graduation is by discipline, and we are missing a lot. There are no complex questions in the world that can be solved by one discipline. We need the others. And that there is not a question that we have to know everything to be able to do that. You have to count on the colleagues with open ears to listen, and then you can do the job. If you are not listening and you are not understanding, you have to ask again what are the idea the other people are saying. So I think that the topic of converging of knowledge is much better than transdisciplinary work, you know. I think it's more idea of you taking different people converging to do something together. I think that this is the good topic for me. Thank you.
Thanks for your comment, Esper. Yeah, but you know, there are a little difference between multidisciplinarity, interdisciplinarity, and transdisciplinarity. The definitions are always complicated, but they have very subtle differences in the way they integrate, because conversions sometimes do not require integration. The conversion need to end in integration to be interdisciplinarity, and if they go beyond that, I didn't talk about transdisciplinarity, but transdisciplinarity is the discipline just disappear and something new, a macro that include everything also is generated. So the thing is how much they integrate or not, and all kind of approaches are good, just working in a single discipline or multidisciplinary research or interdisciplinary. So we just need to understand that there are many different ways to work, and the important thing is that institutions and the scientific community start to understand and recognize properly all this work. If not, we will have, well, we will be discouraging people from doing this kind of research.
venía para verificar los micrófonos. Bien apuntado. Perfecto.
Good afternoon. I am Jose Ortiz in behalf of Dr. Luis Aguilar. I will be trying to keep you enthused, especially after lunch, which is always a tough challenge. So for our first presentation, we have Dr. Francisco Rossi, Challenges for a Permanent Neuroscience School in Latin America. As soon as we can get the connection going, I think we go. Greetings. Could you try speaking, because I can't hear you.
Hello? Oh, can you hear? Can you see? Hello, hello?
Whenever, could you speak a bit louder? But
Hola?
Hola.
Hello? Can you hear now? Yes?
Okay. Go ahead.
Hello?
Yes.
Right? That's
¿Perdón? ¿Cómo es el asunto?
Que se le fue la señal, el doctor quedó expuesto.
Okay.
En cuanto regrese.
Okay. Okay, it seems like we have a bit of a connection problem, in case you haven't.
hello?
Hi there.
Hi.
Hi.
Does it work now? Or?
Yes, now we're connected.
Okay, great. Can you see the screen?
Go ahead, whenever you're ready.
So can I start? All right, just a second. Can I start?
Sure.
Go ahead.
For you.
Thank you. All right. So, first of all, my name is Francesco Rossi, and I'm here representing the executive committee of FALAN, which is the Federation of Neuroscience Societies of Latin America and the Caribbean. So first of all, I want to congratulate the speaker of today for their very interesting presentations and very stimulating, and as well as the organizer of this summit and of course the coordinator of LATBrain. So the organizers asked me to present a talk that is entitled Challenges for the Creation of a Permanent Neuroscience School in Latin America. And this is what I'm trying to do, I will try to do. But before, I thought it was relevant to present at least briefly what is the federation, FALAN. So FALAN was formally created in 2013, of course through a legal statute, and its headquarters are in Santiago de Chile. The idea of creating a federation, of course, occurred well before 2013, when different relevant scientists of our region met and discussed about this idea of congregating groups of neuroscience programs and societies to make neuroscience in the region stronger. So today we have different members, different kind of members, as the Sociedad Argentina de Neurociencia, la Sociedad Brasilera de Neurociencia y Comportamiento, Sociedad Chilena de Neurociencia, el Colegio Colombiano de Neurociencia, la Sociedad de Neurociencia de Cuba, la Sociedad Mexicana de Ciencia Fisiológica, and la Sociedad de Neurociencia del Uruguay, as well as the Sociedad para la Neurociencia del Perú and the Programa de Investigación en Neurociencia de Costa Rica. We are also trying to contact neuroscientists in other countries of our region, so to understand the state of art of neuroscience in these countries and eventually help them become member of our federation. So, as you know, FALAN is a scientific federation member of the International Brain Research Organization, IBRO, and as well as a co-founder of LATBrain, the Latin American Brain Initiative. We work mainly through an executive committee, which is elected each 2 years. We are now at the 6th executive committee, where I work together with Maria Eugenia Pedreira from Argentina, Lina Becerra from Colombia, Deborah Sucheki from Brazil, and Thomas Osandon from Chile. So we have also other committees, as the scientific committee, that is in charge mainly of managing academic activities, which is integrated by members from all the different FALAN societies. And the communication committee, again, integrated by different societies members. We can count on Daniel Sosa, who is our webmaster, in charge of the different technical aspects of the federation. We use a website, we have a networking platform, we use different social media, and we have 3 different email addresses. I left you here one of them, the main one. And we try to send more or less regularly a mail containing FALAN news and activities to our members, and a FALAN newsletter to more than 4,000 contacts all around the world, of course, mainly in the region. So briefly, I would like to share with you the strategic plan of FALAN, which focuses on 3 main areas, which are community building, education and training, advocacy, and engagement. So of course, one very important objective is around community building. So the idea is to promote and facilitate the interaction among member societies and the individual all around Latin America and the Caribbean, as well as out of our region, of course. The main actions are the spreading of Falan and its activities, and the scaling up of our Falan networking platform, which is a platform that emulates a kind of social network. So, for instance, we organized through this platform the Falan Virtual Congress in 2021, where around 900 participated simultaneously. So the second area has to do with education and training. So the mission is include the promotion of neuroscience education and training, of course, and we have two main actions. One has to do with the training program in neuroscience, and as you will know, the next one, the first one, will occur in Chile in 2026, and the Falan Congress, which we consider a crucial platform for sharing cutting-edge research. The next one will be in Chile in 2026, immediately following the school. The third area has to do with advocacy and engagement. So the idea is promote the understanding of neuroscience and scientific activities in general among the general public and policymakers, with three main actions: spreading neuroscience in the region through workshops, conferences, so to enhance public awareness of neuroscience; diplomacy, which is focused mainly in engaging with government and parliamentary decision maker to advocate for the Falan agenda; and social commitment. We, for instance, launched recently a campaign, Falan Women in Neuroscience. Of course, Falan endorsed the ALBA Declaration, and we and organized workshops around neuroethics and neuro rights. So the main previous activities, very briefly, the Congress, as I told you, we organize a Congress each 4 years. The first, let's say, Congress that we consider the first Congress of Falan was in 2008 in Brazil. The first formal one was in Mexico in 2012, then Argentina, 16. In 21, of course, for the pandemic, we had to organize a virtual one through our networking platform, and the last one in 22 in Belém in Brazil. So the next one in Chile, in Santiago de Chile in 2026, we had approximately in these congresses between 900 and 1,300 participants. We organized some academic and or networking activities by ourselves or in collaboration with LATBrain and or LATBrain, as for instance the Falan Roundtable and the LATBrain meeting during the Falan Congress, and an interesting symposium that we co-organized during the last IBRO Congress that was entitled Tackling Global Issues in Neuroscience, where participated actors from other countries, countries from not from Latin America, as well as from Latin America, of course. And then other aspects, we evaluated and managed the LATBrain travel grants and symposia grants for the Falan Congress, and the abstract slots for Latin American students for the Congress of the Society for Neuroscience. So let's go to the main part, permanent neuroscience school in Latin America. So the idea that I had, oops, can you still see? See, oops, so is to present kind of timeline of what the federation has been doing in the last years. So we can say that everything started with an initial meeting in 2023 that we called a school task force meeting, where Falan participated to this reunion together with a representative from IBRO, from IBRO-LARC, the regional committee, Latin American Regional Committee of IBRO. There were members of LATBrain as well as other scientists. So from this first meeting, it emerged, in summary, that the will and the commitment to keep ensuring the high quality training of human resources in Latin America, and that this initiative should be coordinated by Falan, as it is considered a hub of neuroscience in Latin America, due to its structure, let's say. So, of course, Falan had to discuss this idea, the general principle, with Falan members. So we discussed it, and this was approved by all Falan members enthusiastically. And the first important step that we had to do was to reinforce, let's say, the scientific committee, the Falan Scientific Committee. As I told you, it is integrated by representatives from all societies, but we also met with experts in the field, meaning directors, past director, present director, or coordinators of neuroscience school in Latin America and the Caribbean. So what we did, many things, different things, but an important part was to analyze these past and present experiences in organizing neuroscience schools, not only in Latin America, but also in the United States and in Europe, where you know there are very important and traditional schools of neuroscience. So what we understood, part we already knew this, that Latin America is particularly strong in training human resources. In fact, I think Luisa presented the survey where this emerged quite clearly this morning. And we have a lot of different schools, specialized schools, general schools, and very high level. And in any case, we had to identify 3 of these schools: the Ricardo Miledi School, the Latin American Training Program, better known as the Escuela de Neurociencia Latinoamericana, and the Latin America Computational Neuroscience as examples of schools with long history and having formed a lot of different students. And this initiative were fundamental to guide this idea, the Falan initiative. So very quickly again, we analyzed, let's say, the strengths and weaknesses of the past experiences. And of course, we found that these schools played and play an outstanding role in forming students over decades in Latin America, but that the lack of permanent funding caused interruption in some of them, at least, being the main funding coming from IBRO, Grass Foundation, the Society for Neuroscience. So the challenge, of course, is to create a school that can be permanent in time, mainly economically speaking. So to provide continuity to high quality education. So the difficult part is search for collaborations, meaning so what we were doing and are doing and will do is keep contacting potential stakeholders, so in order to build a partnership. At that time, this led to the organization of a meeting that we called the kickoff for the strategic plan of the Falan Neuroscience School, that was held in November the 6th in 2024 in Montevideo, Uruguay, also thanks to the collaboration of the Uruguayan Society for Neuroscience. In this meeting, there were different participants, besides the the executive committee of FALAN, the present, and some of the past members, the FALAN member societies, representative organizer of other Latin American schools, members from IBRO and IBRO-LARC, and other guests were from the Max Planck Institute in Germany, Institut du Cerveau de la Moelle Épinière in France, the Institut Pasteur de Paris and de Montevideo, the Institut d'Investigazione Biologica Clemente Stable in Uruguay, Société Française des Neurosciences in France, and people from the National Institute of Aging and the National Institute of Health, from UNESCO of Paris and Montevideo, Neurobridge Consortium in Italy, LATBrain, of course, Bridge Latin America, and the Latin American Bioimaging Network. So during this meeting, of course, we summarized kind of assessment of the state of training programs in neuroscience in Latin America, focusing on the impacts and the difficulties. IBRO-LARC presented the present funding programs in our region, the school strategic plan, and the idea, because it was well before of the first edition of the school were presented, and then the partners presented the potential strategy to collaborate in our region. And then we had, through the concluding remarks, the signature of a letter of intention, where the participants recognized that establishing a collaborative framework is fundamentally needed to advance neuroscience in our region. And they committed to engage actively with FALAN to seek and implement potential cooperation strategies. So, what now? Present, we are working hard to organize the first edition of the FALAN Latin American Training Program that I told you will be held in Chile 2026. We keep meeting with potential partners and try to define a permanent strategy, at least a short middle term. And in function of the success of the first edition of the school, we are going to organize the second edition. We don't know if in 2026, 7 or 8. So an important aspect that I would like to present is that, as I told you, FALAN has been recognized by other organizations as kind of a hub because of its structure, meaning that it is integrated by neuroscience societies from all the regions. So this initiative can be different, I hope, because there is a regional effort to make this neuroscience school permanent, mainly through the support of the different neuroscience societies that form and integrate the federation. So before concluding, very, very briefly, I will show you the general idea of the first FALAN school that will be held in Chile 2026, and this will be possible mainly thanks to the collaboration of the Sociedad Chilena de Neurociencia and the support of very important institutes in Chile, the Pontificia Universidad Católica de Chile, the Universidad de Chile, the Universidad de Santiago de Chile, and the Universidad de Valparaíso. The organization is, let's say, the classical one with a duration of 3 weeks from 10 to 28 August 2026, and approximately 30-40 students with 3 modules: molecular and cellular neuroscience, system neuroscience, and computational neuroscience. The students will have also the possibility to participate to, let's call them, public outreach events, as the FALAN Congress, that will be from August 31st to September the 3rd. It was organized in this way, so immediately after the school. And the Neurofest, which is a very interesting activity, huge festival, let's say, where actors from different fields, not only from science, but from theater, music, and so on, meet and try to develop activities to enjoy and communicate neuroscience to everybody, to everyone. So in fact, 2026 has been declared the Year of the Brain in Chile, so I hope to see you there soon. And before concluding, I would like to thank you very much for your attention and the organizer for inviting me. Let's say that part of the content of this talk has been published in an interview that was published recently in Nature Communication Biology, which is called Advancing Neuroscience Research in Latin America and the Caribbean. So thank you very much for your attention.
Thank you, Dr. Rossi. I'm sure we have a couple of questions. Okay, then I'll ask you a question. What would be the requirements for such school?
Hello. The requirement for students?
No, for having the school.
For having a permanent school?
Yes.
Or a school?
A permanent school.
Well, the idea is we are trying to convince the relevance of having a permanent school. We already have a different presentation about the difficulty of convincing, let's say, agency to support initiatives, generally speaking. But the idea is to make kind of, how do you say, agreements, formal agreements to support the school, not just once, at least, as I told, at least 3-5 years. So we were working a lot with IBROLARC, but they changed IBRA and IBROLARC. They recently changed kind of the political strategy in general, and of course in Latin America also. So we are going through other channels, and some of them you were listed, were present in this meeting that we had in November 2024. So the idea is, I hope that the difference is try to, the effort is try to find long-term funding. That's the main point, I suppose.
Well, thank you for your input, and then we move on to our next presentation. Oh,
thank you very much.
Sorry for it. I haven't done this in a while. Okay, next presentation, the challenges for the permanent neuroscience, the importance of a Latin American consortium for the study of traumatic brain injury and its long-term consequences, by Dr. Cesar Santana Gomez from UCLA.
Hello everyone, can you hear me? I think so. Hello everyone, can you hear me now?
Welcome, Dr. Santana.
I think so. Okay. So thank you very much for the invitation. For me, it's an honor to be here today at this really important symposium organized by Dr. Luisa Rocha. You see me? I think so. Okay, so I'm working for the Department of Neurology at the Geffen School of Medicine here at the University of California in Los Angeles. And today I will be talking about the importance of building a Latin American consortium for the study of the traumatic brain injury and its long-term consequences. First, I want to begin just saying that I have no conflict of interest to declare regarding to the data or images that I will be presenting in the next slides. The traumatic brain injury, or TBI is defined by the Centers for Disease Control and Prevention as a disruption in the normal brain function caused by bumps, blow, or jolt to the head, or penetrating head injury. It can be generated by many different situations, such as falls, traffic accidents, firearms injuries, or some sport-related impacts, which are really usual especially in the sports that are having contact, like boxing, football players, soccer players, and many others. Depending on the severity of the trauma, the consequence can be very variable. And this is why we often call the traumatic brain injury as a silent epidemic, because in many cases the consequences are not really visible, but they are long-lasting and life-changing. We usually divide the consequences after a traumatic brain injury in 3 different groups. Physical problems, which include epileptiform activity, which should be acute or in the long-term period, loss of the consciousness, sleep disturbance, blurred vision, chronic pain, headache, amnesia, vertigo, and many others. Cognitive problems, which include like memory loss, trouble with the concentration, and difficulty to process new information, or behavioral ones, which include PTSD, apathy, anxiety, impulsivity, depression, or even change in the social interaction with the others. It's important to mention that this is not affecting only the patient with traumatic brain injury. It also impacts the families, caregivers, and healthcare system as a whole. And I think here is a really important key point. If we're able to find some treatment or early strategy to intervene, so we should actually prevent some of these consequences, delay their onset, or at least we should be able to reduce their severity. I think that's why more coordinated research and stronger public health efforts are urgently needed in the field of the traumatic brain injury, especially here in Latin America. As I just mentioned, traumatic brain injury induces severe consequences at the individual level. But when we look at global picture, the impact becomes even clearer. Every year, about 69 million people worldwide suffer of a traumatic brain injury. When we compare with other neurological diseases like Alzheimer's, stroke, or Parkinson, traumatic brain injury remains among the top 3 causes of injury-related death and disability. And the cost to the global economy is around $400 billion every year. Now, if we look at closer, so we can see important difference between the countries. Again, the main causes of the traumatic brain injury are still falls, road traffic incidents, and violence. But the border is greatest in the low and middle income countries like Latin America. But it's important to mention something. There is a very few studies that truly capture the nature of the traumatic brain injury in our region. I think this lack of research means that Latin America is largely underrepresented in the global datasets. As a result, we face serious limitations, such as fewer resources, less visibility, and insufficient evidence to design effective and locally and relevant solutions to treat the people with traumatic brain injury. In other words, I think without research in the traumatic brain injury, so especially here in Latin America, this remains invisible and therefore does not get the attention or priority that it deserves. As I mentioned it before, in Latin America, the lack of data makes it difficult to understand the real dimension of the problem. But if we look at countries where more reliable data exists, such as United States, so we can see the magnitude more clearly. According to the Centers for Disease Control and Prevention, every year there are about 214,000 hospitalizations and nearly to 70,000 dead people related to the traumatic brain injury. That means that every single day, around 586 people are hospitalized and 190 people died because traumatic brain injury. Now, when we look who is the most affected, so the numbers show that older adults over 75 years of age are the most vulnerable group. They are followed by children and adolescents, who are also in the high risk. But these findings are not only in America. This happens when we review data coming from Europe, so we can see the same vulnerable groups at the two extremes of the life, the really elderly people and the really old people. I think this is an important reminder that traumatic brain injury is not just a young man's disease. It can affect people at any age, from children to elderly, and its consequences can be devastating across the entire life. So far I just show you how the numbers looks in US and Europe. But in Latin America, the situation presents a special challenge. Current estimations suggest anywhere between 70 to 360 new cases per 100,000 people each year. Most of these cases come from traffic accidents and violence, which remain one of the major public health issues in this region. The mortality reported about 2 to 3 deaths per 100,000 people per year. But again, as I mentioned it before, these numbers are very likely underestimates, just because the data is incomplete or it doesn't exist. And here takes place the real problem or the real challenge for the Latin America. A region we face delayed access to the emergency care, limited availability of rehabilitation services, and perhaps most importantly, the absence of national registries to track and understand the magnitude of the problem. So all this makes the traumatic brain injury as an urgent but still too often invisible health problem for Latin America. But as I mentioned, traumatic brain injury is a big problem in Latin America and the Caribbean. But some good news are that exists evidence showing that the coordinated action can save lives. This is an example of this coordinated action. So in this systematic review, they present data from 3 different centers that they are treating the traumatic brain injury. They just classify the centers depending on the income level, including one from Monterrey, Mexico. And they observed that the only case that, for example, in Seattle, they have some protocols and registry comparisons and protocols to follow the people after they suffer traumatic brain injury. And this, just having these measures and protocols can reduce the traumatic brain injury mortality in up to 20%. Unfortunately, in Latin America currently lacks this kind of coordinated effort. And this gap in the research prevents us to improving the outcomes for thousands of patients every year. This is why I think creating a Latin American consortium will be so powerful. It will allow us to collect data, standardize data, share resources, harmonize protocols, and foster the real multidisciplinary collaboration across the countries. But why we need a multidisciplinary collaboration? It's important to mention that the outcome of the patients after traumatic brain injury is not only related to the severity of the damage. Many factors like age, brain injury, mental health, and the social and environment aspects, including social support and the access to the healthcare, are so important to improve the outcomes of the patients after the trauma. So to truly understand the long-term consequences of traumatic brain injury, the research must bring together many fields, including neurologists, psychologists, psychiatrists, sociology, and many others. So I think this is why I want to emphasize that building a Latin American consortium is not just an opportunity. I think this is an urgent need for us. So just to show you what is possible, let me share with you a couple examples. One of them is transforming research and clinical knowledge in traumatic brain injury, or TRACK-TBI. This is in the United States, and this initiative brings together 18 trauma centers and has enrolled more than 3,000 patients across countries. The main focus of this initiative is on the discovery biomarkers, prognosis, and outcomes measures, which are helping to define how we evaluate or predict the recovery of the patients after traumatic brain injury. But not only in US, in Europe is another initiative that is called the Collaborative European Neurotrauma Effective Research in Traumatic Brain Injury, or Center TBI. This initiative is even larger. It involves 68 centers across 20 countries with thousands of patients. This initiative is advancing in the field through the precision medicine, advancing image, genetics, and even cost-effective analysis. It's important to mention that these both initiatives are sharing a few things, or some things. One of them is just the use of common data elements and share the information globally, so everybody can have access to this information. I think this level of coordination is allowing to the researchers and clinicians to compare findings, harmonize protocols, and accelerate the discovery of new strategies to treat the traumatic brain injury and improve the outcomes of the patients. I think together these projects are truly reshaping how we study and treat the trauma. And I think a key point here as well is what we can learn from them. So what we can adapt from these initiatives and put in the reality of our region or in Latin America. Of course, the human studies alone are not enough to fully understand the traumatic brain injury. That's why preclinical research using animal models plays a critical role. Over the years, researchers have developed some various animal models to replicate the traumatic brain injury in different species, including rodents, pigs, even genetic models or in vitro models. Each one designed to replicate the specific aspects of the traumatic brain injury. As many other models, so they have strengths and limitations for sure. For example, the rodents are excellent for the genetic manipulation and mechanistic studies. Also, they are very important and really useful for behavioral tests. But their brain anatomy differs from humans. By the other hand, pigs have a brain structure more similar to the humans, making them more useful for translational studies, but they are more costly, and by their size, they are more complex to work with. Finally, in vitro models, so they allow us to precise cellular and molecular studies, but they cannot capture the complexity of the intact brain or the behavior. So there are some of the limitations. However, despite this difference, all these models are essential because they allow us to reproduce the mechanism of the traumatic brain injury and recovery, probably find some biomarkers or early biomarkers for some comorbidities in the long-term period through the EEG recordings, image, molecular, and many other ones. With the use of a preclinical animal model, we are able to, or just preclinical models, we are able to evaluate the therapeutic interventions at different levels and different stages of the disease and understand just tracking the long-term consequences of the traumatic brain injury. This is exactly a thing where the preclinical and clinical research must come together by linking the lab discoveries with human studies. So I think it's the only way that we can accelerate the discovery of biomarkers validation, improving the therapeutic strategies, and gain a deeper understanding on the traumatic brain injury over the time. So I want to show you a good example of how preclinical and clinical research coming together is improving their quality life or the research on the traumatic brain injury. And an example is EpiBio's for RX. So this project is where I have been working since the last few years. And it's a multi-center initiative designed to identify and validate preclinical biomarkers for post-traumatic epileptogenesis by EEG, image, and molecular ones that could be translated to the humans. Let me do a brief introduction. Post-traumatic epilepsy is one of the long-term consequences after traumatic brain injury. This condition affects around 30% of the patients with severe traumatic brain injury. That's why we're highlighting the urgent need of early detection and intervention. That means as early we detect some biomarkers, more early we can start with the treatment of the patients. This project uses just standardized data from different centers, so we are 7 centers around the world, using a specific animal model of traumatic brain injury with the goal of facilitating the development of preventive therapies and improving the risk prediction for the development of post-traumatic epilepsy. I think EpiBios4Rx has transformed the research on post-traumatic epilepsy, providing reproducibility across the centers, harmonizing the experimental pipelines, and validating candidate biomarkers of epileptogenesis. So this project is one of the examples that the power of the integration of preclinical and clinical work, which for sure just can accelerate the discoveries and provides a model that we can follow here in Latin America, probably not just with the epilepsy, with many other comorbidities that are presented after traumatic brain injury. So far, I think I was able to share with you the magnitude of the traumatic brain injury both globally and in Latin America. I think we have some lessons that we need to learn from initiatives like Track TBI, Center TBI, as well as the importance of preclinical models in the study of the traumatic brain injury and the successful example of EpiBios4Rx enriching the preclinical and clinical research. All this together shows how the coordinated multi-center and multidisciplinary research can truly transform how we understand, prevent, and treat the traumatic brain injury and its long-term consequences. I think in Latin America we have the opportunity to generate a consortium for the study of the traumatic brain injury. With this, we should be able to standardize data across multiple centers' involvement, share resources and expertise at different levels, harmonize protocols both preclinical and in the clinic, and the collaboration or multidisciplinary collaboration among neurologists, psychologists, psychiatrists, sociologists, and many or basic science and many other sciences that are involvement in the environment of the people. Finally, I think creating a Latin American consortium is not just a vision. I think it's an urgent and feasible goal. I think the only way is if we work together, we can improve the patient outcomes, generate robust data, and give the traumatic brain injury the priority that it deserves in this region. Finally, I think I have a couple of minutes. So I want to introduce this new initiative, which is the Latin American Association for Traumatic Brain Injury Research, or Alianza TCE. I think this is a great start point for building networks and collaborations that advance in the traumatic brain injury research across our region. This initiative has been led by Dr. Servando Medel Matos from UCLA. He works at the pediatrics department. Dr. Luisa Rocha from Mexico, and myself here at UCLA. And our goal is really, really simple. So we are looking to establish an environment where the interdisciplinary research can thrive and facilitate the generation of knowledge that benefits both the scientific community and the patients affected by traumatic brain injury. As part of this initiative, on September 22nd, we will hold our first virtual symposium entitled Exploring Traumatic Brain Injury from Clinical Impact to Preclinical Research. Please scan this QR code so you will have all the information for registration on the program. But we will be discussing some general and neuropathological aspects of the traumatic brain injury in the clinic. So we will be discussing also the peripheral consequences of traumatic brain injury, And finally, we will be having a really interesting presentation about how the preclinical models can help us to understand the traumatic brain injury. Please follow us in the social network and just asking for more information. I hope you can be there, just sharing ideas and working together in transforming the traumatic brain injury research in Latin America. I consider that initiatives like these are how we move from understanding the problem to creating solutions that truly impacts in the research. Thank you very much. So I am open for questions, and thank you again for the invitation.
Thank you, Dr. Are there any questions? Are we transmitting? Are we zooming? ¿Estamos en Zoom? Ok. Porque no lo veo si hay pregunta. Okay, anyway, thank you again. And since there are no further questions, then we go to Dr. Fernando Cardenas, who will talk about neurodiaspora, collaborating with Latin American neuroscientists outside the region.
Okay, good afternoon. Thank you very much for this invitation. I'm going to talk, and I am sure this will be a very short talk, because there is not so much what we can talk about the diaspora, and we can see an example. It's just one example I want to show you. But first, that I want to say some words about what is the diaspora, how we can conceive the diaspora, and this thing. The very word diaspora. It's not working. Okay, it's something that has little in itself. The word is a little confusing, because you can imagine something like chaotic or not very organized, for example, flight of bugs, insects, something like that. But the meaning of diaspora in this context that I want to show you is something very more organized, with a name, with an objective, with some, we can say, rules, for example. So we can work toward this term. The first thing is to think about two other terms that we can use for understanding the diaspora. The first of them is brain drain. Brain drain is a very common situation in which most people, mainly scientists, go to one country, generally their home country, to other countries. And brain circulation is a certain moment when they return, or they can return, some of them can return to their home countries and create something a little more related to science. With these two terms in mind, we can find some of the reasons because of which some scientists prefer to go outside their countries. For example, this I have four reasons that could be in the root of this migration of scientists. The first of them is the seek for better research conditions. Not always in all countries, and mainly in some kind of countries. We're going to see after this. The conditions are not optimal to create science, to make a scientific career, for example. So many of the scientists tend to go, for example, scientists, I include here students, for example, tend to go outside looking for better research conditions. The same is related to funding. Science, and in particular neuroscience, is a very expensive area, so not always countries has enough funding system for them. A third aspect could be the infrastructure, the need, for example, for laboratories with animal facilities or with other kind of elements, equipments, apparatus. Not always these countries can provide them, so many scientists tend to go to other places. And finally, this is a very important, is the social status. To be scientist not always is the better or the best profession of the best career you can have. Scientists sometimes are seen as those people living maybe on the, we can say, the fringe of the system, people that is not absolutely in a real world as many politicians, for example, see. So scientists tend to migrate from their countries mainly for this reason, and there are other many reasons. For example, familiar reasons. Their kind, for example, have some familiars, parents, even partners going to other places. The brain drain is a very common situation in countries in which it's characteristic low and medium income. So in our region, it's very, very clear, a brain drain in many countries. One of the reasons is for our governments here in most of these countries, science is not a priority. There are always another priorities, social priorities, and it's part of the same life of country. And for this, when science is not a priority, well, scientists not have, don't have many reasons to stay. This is what I was talking about. To be a scientist is not very appealing. And we have some historical examples of what happened when people in this situation on those countries tend to go outside their home countries and establish in another countries. We have, for example, a very well-known case in India, for example. Indian scientists began to this kind of brain drain and went to these three main locations I show here, for example, in the United States, in Canada, and in Great Britain. And after a long time, they began to create what we can call hubs for information. And at this moment, at the present, we can talk about the real Indian diaspora in the sense that they will go there and there find all the conditions to create scientific movements, very strong scientific movements in some areas. For example, these 4 areas are the most known of this development. The same happened in China. They tend to go to another places, for example, Japan or Central Europe, and they there can began this movement in mathematics and physics, for example. So now we can talk about the great constituted, with clear Chinese scientific diaspora. One clear example is what happened with the Jewish scientific diaspora. In the same vein that the other examples, but with a little history, little time history, they began to enter to Australia, to Europa, and mainly United States. And from there, they founded, we can say that, a great scientific movement of help between all of the members of the community in many areas, but principally these areas that are here. In Africa, nowadays, we have 2 really constituted and very supported diasporas. One of them is the African Diaspora Network, and the another is the Carnegie African Diaspora Fellowship. These 2 movements are well recognized and represent a very clear effort, economic effort from other countries. And in the case of Africa, there is no only one country, not just one country in Africa, but they come from several countries, for example, Kenya, for example, South Africa, Nigeria, etc. In our case, here we do not have many clear history as that. We have many brain drain, mainly because of the aspects I told you before, and this brain drain has been not so organized. There are people from these countries in other countries, but in most cases, they even know each other. Diaspora means, for example, help. Means, for example, that I know where my other people from my country could be and how we can make network, we can make some kind of contact. In our region, only Colombia has a very officially constituted example, that is the Colombian neurodiaspora. This is an effort made by the Colegio Colombiano de Neurociencias, as I will show later, and it was some kind inspired in that history I showed. There are some things that characterize this diaspora, 6 aspects that I think are of the main interest, the main necessity to constitute a diaspora. For example, organization. It's needed one organizing committee. It not means people that are permanently in that position, but some kind of organizing committee, maybe temporary, but with clearly established roles, each one. Perseverance, because diasporas have to be maintained in time in order to function. Financial support. This financial support is not something like, for example, grants for investigation, grants for research, grants for publication. This financial support is, for example, to give the minimum that is necessary for this contact between different scientists in different countries. Political support is the need to have a backing organization, preferably something like a neuroscience society in each country. Member commitment, because this is people, and there are people that are in no very stable situation. Not always diaspora members have position, fixed position, and the concept of member refreshment. People come and go in a diaspora. There are students, there are PhD students, there are postdocs, there are people that are maybe initiating their academic life, so people entering, going out a diaspora. These are the things that characterize this movement. In the case of the Colombian diaspora, the backing organization is, as I told you, the Colegio Colombiano de Neurociencias. It's like the Society for Neuroscience in Colombia. They have part of them devoted to help these other Colombians that are in other countries, mainly students, but also researchers or laboratory heads, etc. Here are the 2 concepts of the mission that the diaspora reclaim for themselves, and this, as you can see, could be assumed, could be adopted by any other diaspora in any other country, because it's just the mainly foundations of this. They want to build bridges between Colombian neuroscientists and other regions. They want to create these conditions for academic exchanges, and that's it. Nowadays, the Colombian diaspora has 125, I think, active members. It means that it's a great diaspora, considering that it was founded just 2 years ago, in 2023. The organizing committee are these were the people who established the diaspora, as I told you, in 2023, during the Congress of the Society for Neuroscience, and they have had one more meeting in Chicago last year, and they are planning another meeting. So this was the moment in which the diaspora was constituted. Of course, this has a very long story in the back, more conversations and other things. And the members of the diaspora reside in these countries. Most of them, as I told you, are students, so maybe this is a very dynamic map. Some of them can move from one country to another. Some of them return to Colombia. And of course, here Colombia must be also in blue, because some of the members have already returned to Colombia and are already working there, and from there are in the diaspora. In that picture, is the committee, the organizing committee in that day in, I think it was in November 2023. They were. This is a photograph of Norberto Garcia Cairasco. He is, I think, the pioneer of this movement. He was the first person in talking about the need for this kind of network, for this kind of relationship between different people from Colombia in other countries. He is working in the University of Sao Paulo in Brazil, but many years before even the foundation of the Colegio Colombiano de Neurociencia, he was already talking about the need to this kind of network. It has been a long time from that moment, from now. Only 2 years is the time the diaspora has, but it seems that it has a long future because many of the members are really committed with its growing. And finally here, I show one of them, and each one of them with their position, so you can find them quickly. I think this is very important because this is a model, this is a form of thinking about people making neuroscience, students making neuroscience, or young scientists making neuroscience, that could be modeled and that could help, could have some help between each others, and it could be replied. It could be, for example, copied in another countries. So maybe other countries in this situation could have their own diasporas, and who knows, perhaps someday we could have a network of diasporas, Latin American diaspora, and this situation obviously could help neuroscience, our development in neuroscience, and is part of our Latin way. So this was what I was wanting to tell you. Is as an example, you can find some information in the web page of the Colegio de Neurociencias, or even in the SFN in the program. You can find maybe when and where will be this year's meeting. So this was it. I don't know if there's some question about it.
You're done? Well, thank you, Dr. Cardenas, for your presentation. Are there any questions? Are there any questions? No questions? I have one question and one rhetoric question. How long does one need to stay abroad in order to become diaspora? No, it's a good question. Yeah. Okay, no, address that one because the other one is not an easy question. How long do you have to, no idea? Okay, maybe we should put that into consideration in next ideas. For example, you go to the United States to study, are you part of the diaspora, or you're a visiting student, or what? Okay. So that's important to address. The other question which you may not want to address is the following. Are you talking about Jewish diaspora, or you're talking about Israeli diaspora? Huh? Okay, Israeli diaspora. Okay. Thank you. Okay. The next presentation is by Dr. Oscar Arias, on Aurora digital therapy for the clinical treatment of anxiety and depression. Sí, sí, no.
Buenas tardes, guten Tag, good afternoon. I don't know anymore which language I am talking now. Well, because we care about many things, but health problems are a main problem right now. That's why we are presenting for the very first time Aurora, who is the creation of many people, as you can see in this slide.
We have scientists, clinicians, graphical designers, and one of the most important things, money from a pharmaceutical company who is making this possible today. Of course, we have some disclosure about this. Funding was provided by Psychopharma, who is supporting not only the development, also the technology, and paying, of course, the resources for this application. Maybe after the COVID-19, everybody was caring a little bit more about the health mentality, the health of, not only because of the COVID-19, the lockdown was showing us the relevance for meeting like today, even though we have possibilities now like meeting in Zoom or in other social networks. Nevertheless, in Mexico, anxiety disorders are ranked 7, and the major depressive disorder is the 10. Together are now a burning problem that is going to shock the health system in the near future. And it's because of many reasons. Maybe the most important one is because there are only around 400 psychiatrists in Mexico, in a population that more than 130 million people in this country. So you have to think how you are going to solve the problems in the future. And it is not something that we have to care tomorrow. Now try to get an appointment in any health system, in public system in Mexico, and you are going to struggle to get an appointment with a physician. Maybe you have the money to go to Medical Sur right now, and you are going to have the assistant in 1 hour. But if you don't have the money, you have to wait at least 2, 3, or even 12 months, like is our experience working with patients with Parkinson's disease. If a patient tries to reach a level 3 in Mexico, they have to wait and follow several steps to get an appointment. This meta-analysis is showing that the disorder is more complex than we thought. Anxiety disorders are the most complex disorders. It's not only the behavior of something. This is impacting in many spheres of the person. So that's why in the future we'll have to think how to treat not only depression, anxiety, and other comorbidities. As you can see in this slide, the prevalence of mental disorders are really high and it's really evident across the lifespan. We don't have the possibility to test or to ask small kids, but as far as they are able to respond a test, we can see that this problem is for the rest of their life. And you have ups and downs, and you can fail at any time. And one point, mental disorders are more prevalent in women. But I have to ask you, how many men do you believe they are going to accept that they have a disorder? And it's one issue that we have to confront in the future. They have problems to go to the psychologist or the psychiatrist. Maybe the prevalence is the same, but we struggle a little bit more to go to a physician, or especially if the title is psychiatrist. As you can see, the neurobiology of the mental disorders is really complex. Look, you go from no symptoms, then you cross this threshold to a syndrome, if you don't get treatment, this never stops. If you have a recovery or remission, maybe you can solve the problem and keep your life ongoing. Nevertheless, you can have a recurrence 1, 2, 3, 4 times in your life. So you are never healthy again. And that's why we have to keep something to stop this falling apart and to find a way to have a treatment in your pocket. And maybe we can find a solution for this problem. How many hours do you spend in your telephone every day? According with the average, you are spending more than 4.5 hours. How many apps do you have in your telephone? How many of them are to help you to improve your quality of life? We spend more time in social media, checking emails, maybe something else, but we don't care about our own health. You see, this algorithm is quite complex. As a professor, I don't like this kind of slides. I always complain with my students, it's too much information. But this is the algorithm to treat depression and anxiety, and it's quite complex. You can follow the road here. You have from no pharmacological treatments, if this is mild, moderate, but if it is severe, you have to follow this, starting with some drugs, of course. If this doesn't work, you have to increase the dose, and you have more side effects. If it doesn't work, you have to switch. If this doesn't work either, you have to combine drugs, increasing the side effects. Or you have to find a solution to do something else. So there are others that are more invasive, like electroconvulsive therapy or transcranial magnetic stimulation. Then we have perhaps one solution, that is the digital therapeutics. To better understanding, there is an evolution about therapeutic modalities, going from the 900, from looking for new small drugs, then to biologics. Now, maybe it's a reality, but we don't have access to because it's very expensive. Cell therapy and gene therapy, and recently, digital therapeutics. But imagine you are going to the doctor, public or private, and instead of having a recipe for a drug, you get an app. And you're going to say, what is this? So, this new technology is a way to give therapy intention by a high quality software. By the meaning of preventing the disease, optimize the medication use, or to treat different medical conditions. To do it, you have to follow a lot of many steps for regulation. So it's regulated, of course. For this project, we started early 2023 with a proof of concept for validation. We have to follow a lot of steps. First, to think about how we are going to find a solution for this problem. Then, we were working together with clinicians, scientists, people working in innovation. It was an advisory board and making focus groups. Then we start making a map, a roadmap, of how to make the application. There are many applications in the world, but we have, if you want to use it, you have to pay, and they are now quite expensive. So the next step was to go for usability study only with 15 subjects. Then we get approval by their ethics and research committee to start the pivotal clinical trial. We are at this step now, and we are going to present the data. The next step is to start with the multicentric clinical trial. The aim is to recruit 164 patients around Mexico in different centers, and then go for the approval as a medical device software for Aurora. But who else to show you how this app is working than Aurora? Can you help me with the play, please? Allá arriba, ¿podrían apoyarme con play? Sorry, this is not translated to English.
Me encanta saludarte nuevamente. Te felicito, porque el hecho de que estés aquí ya es un gran paso para sentirte mejor. Para que tengas más claro de qué se trata esta aplicación, quiero explicarte un par de cosas fundamentales. La primera es que aquí aprenderás nuevas estrategias para regular las emociones intensas sin huir de ellas ni negarlas, sino aceptándolas para así encontrar la forma de adaptarte mejor a las situaciones que te las detonan. Viéndolo de otra manera, es como si construyeras una relación más sana contigo mismo. Para tener cimientos fuertes, es necesario que identifiques las razones por las que vale la pena hacer cambios en tu vida. Estas razones serán tu motivación para llegar al final del tratamiento. Tendrás un mayor conocimiento de tus emociones al identificar la relación que hay entre lo que piensas, lo que sientes físicamente y cómo actúas ante ello. Te enseñaremos una estrategia para observar tus emociones sin juzgarlas y así puedas ser más comprensivo contigo mismo. Te darás cuenta de cómo lo que piensas influye en cómo te sientes y qué hacer para que tus pensamientos sean más equilibrados. En este punto, te pediré que comiences a actuar para acercarte a tus emociones en lugar de alejarte de ellas. También te explicaré cómo las sensaciones físicas influyen en cómo vives tus emociones y qué puedes hacer para estar más cómodo sintiendo lo que sientes. Llegará el momento de poner en práctica lo aprendido y decirte qué es lo que sigue para ti. Siguiendo con este ejemplo de la construcción, imagínate que tú eres el arquitecto que dirige la obra y necesitas llevar un registro de todo lo que sucede para poder identificar los avances semana a semana. Para que esa especie de bitácora sea más apegada a la realidad, no se trata de decir si la edificación está fea o bonita, o solo escribir las cosas malas que pasaron, sino registrar lo que sucede en todo momento, pues teniendo la información de una manera objetiva, será más preciso identificar los avances del proyecto. Es por esto que en cada módulo introduciremos registros específicos que han sido diseñados para ayudarte a practicar cada nueva estrategia de modulación emocional. Es muy importante que realices cada actividad y le dediques tiempo, pues de ello depende que vayas sintiendo un avance a lo largo de la aplicación. Además, hay 3 registros que usarás durante todo el programa. La escala de depresión y la escala de ansiedad te permitirán anotar la frecuencia con la que sientes ansiedad o tristeza y cuánto ha interferido en tu vida cotidiana. La escala de emociones positivas te permitirá identificar qué tan a menudo experimentas alegría o entusiasmo. Estos registros deberás llenarlos semana a semana en la sección Registros de esta aplicación. El sistema te dará una puntuación semanal que se reflejará en el registro de progreso, donde podrás observar los cambios a lo largo de todo el programa. Sé que con solo pensar en tener que registrar todo lo que sientes todas las semanas puede resultar abrumador y que al principio lucharás contigo mismo, pero créeme que en poco tiempo resultarán más fáciles y sobre todo gratificantes, pues podrás ver cómo vas mejorando semana a semana. Gracias por verme hasta aquí. Te sugiero que al finalizar este video vayas a la sección de registros para que te vayas familiarizando con cada uno. Gracias y felicidades una vez más. Nos vemos en el módulo 1.
So, here's the architecture of the app. It's like your Netflix every day. I know that you can go through several sessions. So you have to start with Modulo 1, then to Modulo 8, of course. And with this QR, you can download the app and give us some comments, of course. So you're going to see something like that. It's a digital experience. Maybe it's completely different than attending a physician, a psychiatrist, or a psychologist. But you are going to analyze what happened with the subjects now. So we have a pilot experiment to see the usability. Many of, if not all of them, complain about the excessive test density, maybe because there are no researchers and they are not reading a scientific paper. Then they complain about limited interaction feedback, that this can be solved by a call center 24/7. They complain also about some navigation inconsistencies. The problem with the app is that it's running with internet, and as you know, in Mexico we have some problems with that. If you try now to use your telephone, maybe you have some problems because of the structure of this building. So this test allows us to improve the app, and then with 38 users, by the end, we get the threshold of 78.5. Now the users are more happy with the usability. They also find by another application the functionality, engagement, information, and also mention that the application was beauty. In addition, we have some data about it was easy or not, it was relevant for the daily life, and if at the end they find a solution for the health problem. So next step was to confront the app with the clinical trial. We started with 46 subjects, nevertheless only 38 finished the study. We have here all the parameters. The age was 31.2. From 18 to 45, most of them were female. The education is a point. All of them, if not most of them, were at the university or higher, even some with master or PhD degree. The diagnosis was, well, the diagnosis is different in this case. So you will have some cases with anxiety only, depression only, and most of them with anxiety and depression. How we mentioned, this is a mixed disorder. So we have other measurements, but it's not relevant to mention here that we care about the treatment, because the treatment, the pharmacological treatment, was not the aim of this step. As you can see, we can see some improvement related to anxiety scores and depression score. Nevertheless, this is not the main aim of this study. We are still in the usability part, and the next step is going to be the clinical trial. We can find some correlations, and one of the most important is, the more you use the app, the better the improvement of the symptoms are. So this is not something that we didn't expect. So we are expecting in the future to find a better way to engage the user to this application. That's why in the clinical trial, we are not going to test the app against the in-person treatment. So next step is the multi-center clinical trial. We're expecting 164 subjects. The design of this phase 3 randomized multi-center open-label clinical study to evaluate the efficacy and safety of Aurora is going to be like this. We have one arm with the treatment. One important thing is all the subjects are going to be naive, never with treatment, neither psychological treatment. If the treatment doesn't work, we are going to change the treatment. And in the arm B, we have also the treatment. We can shoot the two. And in addition, Aurora, they are going to have the application, and there is a follow-up for 12 weeks. And at the end, we are going to check different ending points. So the outcome is to see how Aurora is improving the evaluation from the psychiatrist and of course from the arm for the measuring of anxiety or depression. So to do this change in the way we are treating mental disorders could be maybe not that easy for many users. We are really close to the physicians, we are really close to the psychologists, to the psychiatrists, so we have maybe to think what is going on in the near future. And that's why we invite you to download the app and test it and get some feedback from you. You have any question, I will be happy to talk with you.
Is it possible to download the app?
Yes, you can.
Okay, anyone wants to download the app? No, I teach psychopharmacology, so this is one interesting challenge. Yes. Question for you: can this be used for other conditions?
Well, we have to adapt, but well, support for anxiety and depression would be the thing about those disorders is the comorbidity. So, in principle, it can be used.
Well, let me get the app and then
you can get it.
Usually one does not think about being depressed or anxious, but I'm sure you know at least. Usually one doesn't think of oneself as being depressed or anxious, but I'm sure you know at least one person that will fulfill the roles. For example, depression is 20% of the population in the United States has bouts of depression. That's a lot of people. So.
Oscar, what is the relationship between anxiety and depression? So one leads to the other or are independent
health issues?
Well, the physiopathology is different. They have some, well, at least by using functional magnetic resonance, you can see that they share some structures, but the neurochemistry and the neurobiology is different. But if you have anxiety, the risk to have depression is higher than normal people, and the other way around.
So they have a connection with the vagal nerve? Is the vagal nerve related to both conditions?
Well, curiously, there is a treatment that is an electric stimulation through the vagal nerve, and both are improving the symptoms.
Thank you.
Thank you. I have a question. Is this application only for Mexicans, or does it have influence about cultural aspects? For example, neuropsy was adapted for Mexican population.
Yes.
Because it was different for other populations.
Yeah, this is the same thing for the app. It's going to be now for Mexicans, and the manuscript is submitted, and some comments from the reviewers were about that point, and it's true. So we have different cultural or meaning for words. So if we want to use it in another country, it has to be adapted.
Thank you.
Are there any difference between males and females as far as the response or sensitivity of the app?
So now we don't have any conclusion because the number of females were higher than the male. So who is more engaged to the app? Curiously is the men.
No, the thing is I have to point the mic away from you.
Well,
thank you again. And for our next presentation, we have the International Brain Initiative, Advancing New Partnerships for Brain Health, by Dr. Julie Isles.
Good afternoon.
Good afternoon.
Let me get my slides going, and we will do a check. How does this present? Does this look all right? Colleagues, can you see my slides?
Hello? Yes, we're hearing you.
All right, very good. And you can see my slides as well?
We're good.
Excellent. Well, let me just say how delighted I am to be joining all of you for this Latin American Brain Initiative Summit and the opportunity to speak with you about the International Brain Initiative and new partnerships that we see through this consortium for global brain health. I would like very much to acknowledge our funding organizations, primarily from Canada at this time, the Fonds de Recherche de Québec and the Canadian Institutes of Mental Health, Neuroscience, Mental Health and Addiction, as well as the Brain Minds Consortium from Japan, all who have contributed to the ongoing efforts of the International Brain Initiative. And of course, deep thanks to Dr. Luisa Roja, LATBrain Executive Committee member, and to all Executive Committee members and the staff of the International Brain Initiative. So my objectives for the next 20 minutes or so, and I hope to leave time for discussion, is to introduce the International Brain Initiative Consortium to you, highlight examples of the robust activities of the IBI, International Brain Initiative working groups,
use the successes that we've had to date as a platform for discussions with you about future directions specifically for LATBrain, and indeed to engage in an open discussion. So the first generation of the International Brain Initiative was declared in Canberra, Australia in December of 2017, and the first generation lasted 5 years. The declaration suggested and recognized major national or cross-national neuroscience programs that spanned not just projects and programs, but major initiatives across or within countries, and that the effort was so large and complex that the need to coalesce the efforts of these major initiatives was brought together into the IBI. The idea was to form an alliance, to take stock of our efforts and resources, and also to declare where possible where common outcomes and priorities lie. And at the end of the day, to maximize the value of and return on public investments in neuroscience research. At the time, in 2017, the International Brain Initiative comprised 5 regions of the world: Canada, from where I'm joining you, the United States, Europe, China, South Korea, Japan, and Australia. We now are in a second and very exciting generation of the International Brain Initiative that began in 2022 under my leadership with my co-chair, Dr. Tony Hanan from the University of Melbourne in Australia. And our motto really is captured so beautifully by this African proverb that was quoted to us by Dr. Alfred Numanchi, the executive member from the African Brain Initiative. And that is, if you want to go fast, go alone. If you want to go far, go together. And indeed today, the International Brain Initiative encapsulates all continents of the world with 11 representatives from 11 regions of the world spanning all continents. And you see them here. LATBrain is now representative, New Zealand, as well as Finland, adding to the prior 7 that I named earlier, and of course, Africa. So what's the vision of the International Brain Initiative? It is to engage in conversations about global neuroscience, scholarship and innovation that is free of geopolitics and borders, and that is really catalyzed by critical engagement. And as well, it's to provide a platform for active dialogue and exchange, and a hub of gravitas that enables regions of the world that wish to coalesce into a national initiative to come to us and have our support, and then become part of the community of IBI leaders with support eventually from their own governments. And LATBrain, for example, is a perfect example of where we have embraced LATBrain's efforts and hope that we can assist LATBrain in garnering government support for your neuroscience efforts in your region of the world. So the initial declaration, in addition to really coalescing and capitalizing on existing resources, was to recognize how society can be impacted in cooperation with one another and across all sectors involved in neuroscience: academia, government, industry, as well as citizen scientists. We add to that our continuing commitment to the challenges and the continuing data challenges of volume, sharing, and science, and how all sectors really have to deal with the question of data sharing and tools in today's complex world. And we also recognize quite openly and actively the importance of ethical considerations in all aspects of the neuroscience endeavor, from animal models all the way, cells, animal models, all the way through human interactions and engagement. So right now, the International Brain Initiative has 7 platforms and working groups, and they're listed here. Data sharing, recognizing that priority. Emotional brain, led by South Korea. Brain mapping, led by China. Neurodiplomacy, led by Africa. Cross-cultural issues, led by Canada. neuroethics led by Europe, and education and outreach. And let me give you a few examples from 3 or 5 of these working groups to show you really the kind of work in which we are engaged. The emotional brain, led by Dr. Jawok Suk in South Korea, is focusing on multi-scale knowledge sharing around the circuitry and molecular brain level mapping, integrating post-mortem brain data and clinical information, as well as animal models to identify biomarkers and therapeutic targets for major mental illness. And whoops, forgive me. And high-resolution transcriptomic analysis and translational neuroscience for mental health. And this slide is thanks to Dr. Jawok Koo, who sits on our executive committee, and to whom I'm very grateful. The deliverables from South Korea have been tremendous. They have hosted an annual Daegu conference, an IBI summit. They've delivered postmortem brain studies in depression and schizophrenia, brain studies in alcohol addiction, and indeed they continue with their efforts in translational research and transcriptomics linking depression in animal models. The ongoing work of South Korea is to expand the transcriptome studies across disorders, continue with cross-species analyses, multi-scale platforms, and so forth. And the opportunities that South Korea sees is the synergistic relationships that will lead to shared data repositories and extended research to include digital therapies as well as neuromodulation. And they receive ongoing support from their government's strategic investments in mental health solutions, and they continue to provide tremendous leadership in the emotional brain research group to the IBI. Neurodiplomacy, as I mentioned, is led by Dr. Alfred Nemenshi, shown here. You can see my cursor circling him. He is here engaged in some cultural activities in which the IBI executive delegation participated in China in November 2024. Through the efforts of Dr. Nemenshi and the African Brain Initiative, the Yaoundé Declaration was published and accepted by the UN as a call for prioritization and an imperative of healthy brains for the African continent. And as you see, the Yaoundé is based in Cameroon, where Dr. Nemenshi operates as also a neurologist as well as an executive member of the IBI. The cross-cultural working group that I've had the privilege to co-lead with Dr. Melissa Perrault at the University of Guelph in Ontario in Canada has been tremendously active. We have, our goal has been to share and deliver best practices for research with Indigenous people, not only in Canada, but around the world, explore translational methods and care in the neurological sciences, promote the integration of Indigenous and Euro-Western ways of knowing, learning, and doing in neuroscience, and also to provide guidance on Indigenous capacity building in neuroscience. We published a big perspective piece in Nature in February that was called Two-Eyed Seeing, and our focus was to really explicate how Euro-Western and Indigenous ways of understanding mind and brain and health and disease can coexist with biomedical explanations. And we felt very privileged to be able to share this. We were 11 authors on this paper, including Rudy Taylor Bragg in Australia, who provided this magnificent illustration depicting two-eyed seeing, where the two eyes of traditions and biomedicine can come together effectively. The cross-cultural working group has been deeply engaged in addressing mistrust in neuroscience. Many of you may be familiar with some of the violations of trust that have occurred in the brain and health arena, global health arena, involving the Mazatec people, Havasupai people, in which genetic data were misappropriated without consent for explorations not only of disease, of diabetes, but then of genetic disorders around mental health. Violations around the Sand people led actually to the Sand Declaration for Research Ethics in Africa, a major accomplishment for that region, and the similar violations around the Maori people. And in fact, the Maori people were discriminated and stigmatized when what was called the Maori aggressive gene was identified. All these things have been in focus for the cross-cultural working group of the IBI. As I mentioned, we've been working on building capacity and promoting ways and strategic initiatives for building capacity among not only indigenous people, but also marginalized, historically marginalized people, those who don't typically have resources to conduct research, have access to faculty and mentors and so forth. And this is a paper we recently published in Nature Neuroscience. We also talk about capacity building in a book that the author group of the Nature paper published just recently. It came out in June 2025 called Cross-Cultural Perspectives on Mind and Brain. And we have a podcast in Neuroethics Today that also talks about the initiatives of this cross-cultural working group for the IBI. Our ongoing work is to continue to promote safe, diverse, and holistic and indeed decolonized epistemologies, methods, and workplaces in neuroscience through our publications and advocacy. And we are now moving our work into a series of panels, we hope, within the UN system to bring, for example, topics around AI and the rights of indigenous people to their attention with good constructive solutions and frameworks for moving forward. Our outreach and education group has been working on training models to create global brain scientists, again, always respecting the cultural differences between neuroscientists across all regions of the world. They're developing some educational resources for teenagers and also culturally adapting first response training for mental health. And the app that we just heard about is a very interesting way forward in that regard. The deliverables to date have been to collect some pilot data. There's a social media workshop planned in the mental health arena for Hyderabad for September this month, and educational resources we're expecting in October to February at the beginning of 2026. So what does the future look like for the IBI and LATBrain and the exciting initiatives we have that we have to look forward to? They fall into three categories at least: exchange, cooperation, and elaboration. In terms of exchange, the IBI is the host to a fellowships program in global exchange that is now available in Canada, Germany, China, and South Korea. There are 16 fellowships available for early career investigators. These are postdoctoral fellows and faculty at the assistant professor level, and we are discussing and seeking new opportunities for fellowships in the countries you see here listed at the bottom of the slide, and we sincerely hope that we will be able to create and host IBI fellowships with colleagues in Latin America in the near future. Cooperation involves so many of you in Latin America, in Mexico, in Chile, as well as with a group called World Women in Neuroscience that has really had a focus on under-resourced women in the world, including in Latin America. We've done a tremendous amount of mentoring for women from Latin America in neuroscience, and we look forward to advancing, developing, and transforming the landscape in Latin America and other regions of the world through this organization. And finally, our elaboration is to continue to be present and visible and grow. We are very excited to be joining you in Santiago in Chile at the Falan Congress in 2026 that we heard about at the very first talk of this afternoon's session of the summit. And we are very excited as well to be presenting a number of initiatives of the IBI at the 80th session of the UN General Assembly in New York City at the end of this month. And with that, I thank you for your attention. I know it's been a long day for you, but I would welcome your comments and questions at this time. And again, thank you so much for inviting me and giving me the opportunity to share the exciting initiatives of the International Brain Initiative with you.
Are there any questions for Dr. Isles?
Perhaps I can ask the audience a question, if I may, and that is, what do you see as opportunities for LATBrain to participate and prioritize in terms of global neuroscience? Is it people? Is it topics? Where can the IBI be most helpful to your initiatives and priorities and values?
Hi, Judy. This is Luisa Rocha, and thanks a lot for your participation. And I wonder, how do you see the collaboration between Africa and other countries? Because, well, in some way, we are regions, Africa and Latin America, with low financial, big political problems. We are very similar in many ways. However, Africa established a good collaboration with IBI since a long time ago, and as far as I know, you have very good publications with Africa in very good journals. Then I wonder, what is your opinion about this issue?
Oh, that's a wonderful question, Luisa. Thank you so much for asking it. I think Africa has provided us with a model of resilience that perhaps no one could have expected. And the effort and success in bringing that Yaoundé Declaration forward that speaks to the importance and prioritization of brain health for Africa is an example and a model for perhaps what LATBrain can do. Perhaps there should be a LATBrain Declaration for Brain Health that will bring the attention of the UN to LATBrain, of UNESCO to LATBrain, of the World Health Organization to LATBrain, and of your own governments to LATBrain, and help build more resources for neuroscience in your region of the world. So I actually would suggest that perhaps we can use the expression low-hanging fruit, but we know that it's never low or easy, is to begin to work on a declaration for Latin America and see how that goes. And we heard about educational resources in one of the talks this session. We heard about apps in this session. I think all of your individual efforts can coalesce into a single declaration that could be indeed very powerful.
Yes, probably we need a very good dialogue among all the initiatives, because we have to know what are the resources in each region. And probably a good dialogue can be carried out in a meeting such as the one you have in China last year, such as the one you are going to have in South Korea this year. In the future, probably you can consider a dialogue, a meeting of IB in Latin America.
Well, we would be thrilled to come to Mexico, and we're coming, of course, in San Diego in September, and perhaps that dialogue could take place then. The other possibility is to engage someone who could actually do research on what are the resources that exist within the LATBrain network, or perhaps you know that. Perhaps IBRO could help us to do the research to know what exists, what the strengths are, what the priorities are, and where the gaps exist, and then proceed in an evidence-based way. The Yaoundé Declaration was really the brainchild, without evidence per se, of Dr. Namanchi, but I think proceeding in an evidence and a scientific way would be very strong for the Latin American region. So however the IBI can help, and I think perhaps we can go forward with a proposal to IBRO to also to assist. That could be a very interesting project together, and could actually form a new working group for the IBI focused on Latin America.
Thank you.
Well, I want to thank you again for participation. Look forward to further interactions.
It's been a pleasure. Thank you so very much.
Thank you. Bye-bye. Okay. Are there any additional questions? Well, if there are no further questions, then I'll thank you for being here. Good.
Señora, esto no es un velorio.
A ver, espérense tantito, por favor. Le pediría a Daniel Olazábal que dé unas palabras finales, todavía están los presentes, por Zoom. Ándale, Daniel. Sí, ya para cerrar. Allá arriba.
¿En español o en inglés? ¿Español? Español. ¿O en inglés? Okay. I was asked to thank you, the people in online, for participating in this first summit of the LATBrain initiative. We really appreciate your participation, your questions, your comments. We invite you, if you have not yet get involved in the activities that LATBrain organized, that we invite you to join us. Invite your institution to participate in the LATBrain, and tell everybody, if you are a student, tell your group of investigation. If you are a teacher or a professor, please spread the news. LATBrain has been working to integrate Latin American scientists in brain science for already 4 or 5 years. So if you communicate and tell people and institution about our organization, is probably going to get stronger, and we are all going to be to get benefit from that. Okay? Thank you very much.